نخلق بيئة جذابة وجيدة ورائعة في وطننا. كيف نكون فخورين في وطننا؟ كيف وطننا يكون جزء مساهم في تنمية وحراك هذا طموح
أنشئ المركز في البحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتوفير عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية. شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر في تستهدف بناء جسر يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، وتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
الحين كذا يطلع صوت.
نخلق بيئة جذابة وجيدة ورائعة في وطننا. كيف نكون فخورين في وطننا؟ كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونستطيع نتقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
جذابة وجيدة ورائعة في وطننا. كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونستطيع نقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى. كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، وسنقدم نقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، وسنقدم نقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980 تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، نقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، نقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجنائية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
كيف نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، ونقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية.
السلام عليكم ورحمة الله وبركاته. خلينا نقول أول شيء بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه أجمعين. On behalf of the chairperson, scientific committees, and the organizing committees here, 22 conference, hosting this day, which is themed under hematology and oncology. So I warmly welcome our dear guests and speakers and audience for this day. We will be starting our session here, which is themed under the hematology and let's say the benign hematology. It will be ranging from clinical trials all the way to the bedside of developing biomarkers. The moderators for this session will be Dr. Ahmed Barifa and Dr. Sani Bahashwan. Faculty of Medicine, King Abdulaziz University. King Abdulaziz University is also an adult hematologist, consultant adult hematologist at the King Abdulaziz University Hospital. Dr. Ahmed Barifa is also the president of the Saudi Society of Hematology, is the head of the Hematology Research Unit at the King Fahd Medical Research Center. Dr. Thani Bahafwan is associate professor in hematology at King Abdulaziz University. Is also as a consultant adult hematology at the King Abdulaziz University Hospital, board member of the Saudi Society of Hematology, as well as principal investigator at the Hematology Research Unit here at King Fahd Medical Research Center. So I welcome the moderator to the stage. We will be having 4 sessions this day, and each session has 4 speakers. The first session will be in their hands, and I know it will be in good hands.
Thank you, Dr. Majid. On behalf of Dr. Salim and myself, we welcome all of you in attending the first session. It's my pleasure to introduce our guest and our keynote speaker, Professor Ahmed Al-Askar. Professor Ahmed Al-Askar is the Executive Director of King Abdullah International Medical Research Center, KMARC. He's the President of the Saudi Society of Blood and Marrow Transplantation, SSBMT. He is a professor and consultant of adult hematology and stem cell transplantation in National Guard in Riyadh. He is a renowned global and national researcher and leader in developing all new biotechnology in the kingdom. He will discuss with us today the clinical trials in hematology in Saudi Arabia, challenges and opportunities.
Bismillah, first of all, thank you, Anwar, Ahmed, Dr. Salim, again for being this. I will talk about trials in hematology and how to put it together. Of talking about trials, whether in hematology or other areas, and just to give you an idea where we stand at the Kingdom of Saudi Arabia and what's needed to be done. And there are lots of things needs to be done, and hopefully through this gathering, we can come up with ideas and suggestions and plans to move forwards to enhance this setup. So in my presentation, I'll take you through what's the impact or importance of clinical trials. So why do we talk clinical trials? What's the landscape of clinical trials in the kingdom, globally and locally? And what's the status of the hem-oncology trials, local statistics, the current opportunities, and how we attract more clinical trials. And I will introduce you to the Saudi Clinical Trial Enterprise, CTE, and how we, again, as hematologists, put the hands together, maybe through the Saudi Group of Hematology Research. All right, so for the clinical trials, when we talk clinical trials, I'll take you through initially some definitions, so we know what we're talking about. We all aware of phase 1, phase 2, phase 3, phase 4 clinical trials, but let's think about it differently now, and think about it interventional versus non-interventional clinical trials. And when we say interventional, that means trials that you need to study an intervention, whether it's a medication or a medical device or a procedure. So you have an intervention, and these needs to be done either through phase 1, where you assess the safety of the intervention, or phase 2, where you assess the efficacy of the intervention, or phase 3, where you assess the intervention versus standard of care. Now, this is on one side. On the other side, there is the non-intervention, which I see that we are distracted by the non-interventional more than the intervention trials. You will see the pharma industry will approach you, many institutions actually. However, they will approach you with non-intervention clinical trials, with post-marketing, observational, chart review clinical trials. And I see this as distracting because it will be counted as clinical trial. However, the impact is not as huge and as important, and it's not part of the development of the medications as for the intervention clinical trials. Of course, the most difficult will be phase 1 clinical trials, and you will not see big companies come to you with phase 1 clinical trials. Because phase 1 clinical trial needs expertise, needs a whole setup, needs specific accreditation, and because it's critical for that medication that took 20 years to develop, and then it could fail at phase 1. So that investment, which is probably come to, on average, $1 billion, could be lost at phase 1. So the phase 1 clinical trials is not easy to attract, and the pharma industry will not come to you because they will go to specific institutions that they get used to and they trusted already. So let's leave the phase 1 for the time being and focus on phase 2, phase 3, because they are again part of the development of clinical trials, and it's important that we get involved in these type of clinical trials. And then you get the classification whether it's a sponsored clinical trial, and most of the clinical trials that we have are sponsored, or in the world globally are sponsored, and the investigator-initiated clinical trial probably would not exceed 20% of the all clinical trials. So you would see the pharma industry leading clinical trials because they are very costly, and it's like investment. So you will not see non-profit organization, for example, able to afford to do critical clinical trials, especially the phase 1, phase 2, phase 3. So it's by default that we need to align with the pharma industry to attract sponsored clinical trials in the country. And that will lead us, again, to develop our own investigator-initiated clinical trials later on. So, again, why do we talk clinical trials? What's the importance? Why do we need to care about clinical trials? Now, there's a tremendous gain at various levels, whether the patient, or the physician, or the hospital, or the country. Now, if you look, we are big time missing out, actually, Saudi Arabia. We are well developed in the healthcare in general. We have the best hospitals that's comparable to the best hospitals worldwide. However, when it comes to the clinical trials, we are way behind. And that's a huge gap that we are not aligned with the infrastructure and the capabilities we have in the country. So what's the gain at the level of the patient? The patient will get early access to new treatment. And I would pause here because maybe because we are a well-developed country and we have ability to access the newest medication worldwide. And any medication that already approved, we can easily import it to the country. And that in itself is good. However, at the same time, is sort of anesthetizing us, not to involve in the clinical trials that offer this medication 10 years ahead of time, 10 years before the approval of these medications. So we have many patients that we run out of options, therapeutic options for them, and they would seek to alternative medicine, trying to get help. However, there are medications that's already in the clinical trials that these patients do not get access to, and it is for their benefit, again, to get access to these new medications that in 5 years or 10 years will become standard of care. So that's a big loss for the patients. The patients will go to alternative medicine, whatever others, and not to go to scientifically processed options. The patient also will get closer medical attention and medical monitoring. When he is in clinical trial, the patient will be monitored for any trivial symptoms. It will be captured, reported, whether it's related or not related to the intervention. Even car accidents, God forbid, if it happens while patient on medications, will be recorded, captured, whether it's related to the medication or not related to the medication. So the patient will be monitored closely, and that's in itself is a benefit for the patient. And there is a potential health improvement. So the patient, for the physician, there is professional development, there is academic recognition, being able to lead clinical trials. There is financial incentives, and it could be very rewarding financial incentives that comes with this fund. And there is, of course, the publication opportunities. For the hospital, there is increased funding that comes to the hospital with any clinical trial. There is enhanced reputation of the hospital, a better infrastructure and research culture, improved patient care in general, and the quality of care gets improved because most of these, again, clinical trials, it's sort of like quality improvement projects. For the country, again, it's a huge gain for the country. There'll be improved healthcare innovation, early access to cutting-edge medicine, especially when the medication is not yet covered by insurance companies. And again, here I would suppose, in many other countries, new medications or medications that's not approved yet will not be covered by medical insurance. So the only way for these patients to access these medications or new innovation will be through clinical trials. And that's why we see abundance of clinical trials in the Western countries and other countries, while it's not as much in here. Of course, the economic impact is huge, and the next slide will So if you see this now, I'll take you this slide. I don't know if you see the point. If you see the right side, if you see in the right side, the global pharma spending globally, it's $100 billion. It's a huge fund that's spent on development of medications. Now, what's the share of, I would say, first of all, the share of United States of this fund is 30%. So 30% goes to the United States. How much goes in Saudi Arabia? It's the bottom, currently 0.05%. So it's negligible, I would say. Now, how do we enhance this? So if we try, so how do we enhance it? Oh, So how do we enhance this to 1%? And that's what we will talk about now. And that's something. This is an initiative that we are working on to enhance clinical trials to get at least close to this 1%. And if we do this, the impact will be $1 billion revenue to the country. There will be 10,000 patients will get access to medications as early as possible before even the medication is approved. And we'll get like 4,000 or plus jobs created related to clinical trial and research. So this is a very important aim that we try to achieve, and I'm sure with the ecosystem that's evolving, we will get there, inshallah. This is another slide to show you, what's globally, what's the situation. Here there's towards the research, and here's towards the consumption. So there are countries who just consume medications, and we are one of them. And we're not participating as much as we consume in the development. So this is a larger zoom of this one. So if you look at, for example, Poland, Belgium, Netherlands, they are not just consumers. They get revenue more than what they consume. They get revenue from clinical trials more than. So all of these countries are on the side of the research and revenue generation. While on the other side here, and here in Saudi Arabia, we're just consumer. We're not trying to get funds out of pharma industry to the country. So all Middle Eastern countries are just consumers of medications. So how much do we consume? So this is, again, to show you how much trials per million people. If you take United States as the standard, there's 540 clinical trials per 1 million population, while it is only 15 or maybe less in Saudi Arabia per 1 million. And look at some other countries are way even above the United States in terms of attracting clinical trials. And here, if you look at the economies of certain countries that's close to Saudi Arabia, this is the GDP. Spain, Turkey, Poland, South Korea, and look at the clinical trials. and here is Saudi Arabia. So we're not as much as, as supposed to be aligned with our GDP in terms of contributing to clinical trials. So I hope I convinced you where we stand and how globally the landscape and the market of clinical trials. Here again, on the other hand, there is global, I would say, call from regulators that are taking active to ensure population diversity. So our population are not represented in clinical trials. That means our ethnic group are not tested in the clinical trials. Most of the populations that get enrolled in the clinical trials are of European ancestry. So this red zone is the European ancestry. So most clinical trials actually represent what happens in the European ancestry, not in our ethnic group. So there we have our genetics, our culture, a different setup, and we're not sure of these medications that the clinical trials represent us well. So we need again to enhance our presence and diversity in the clinical trials, and the pharma industry aware about this, and they are coming to the Middle East, and they're coming to Saudi Arabia again because they are requested to have diversity of the population. All right, now focusing on hematology and globally, what's the magnitude of hematological clinical trials? Here you see the lymphoma. There's like more than 300 clinical trials happening globally now in lymphoma, and these are clinical trials. And there's more than 8,000 sites, again, active in clinical trials in lymphoma worldwide. However, none in Saudi Arabia. On the other side here. Myeloma is the same thing, like close to 300 clinical sites, more than 10,000 population of sites, again, active in multiple myeloma. And we have 3 sites here, 3 studies and 3 sites active in myeloma. So there is a big room improve and to enhance this. Now, what's the situation now here in Saudi Arabia? And these data from Saudi NIH, we have improved. So having said this, in the past few slides, we are improving. And we have a setup now with the enhancement and the push of the Saudi NIH and the awareness among clinicians and hospital administrators. We're improving. Last year, 58 clinical trials in the country. Improved in 2024 to 82 trials. So that's 40 or 41% increase if you take it as statistics. And 21% or 22% of these are in hematology oncology field. And this is from the Saudi registry, Saudi clinical trial registry, the Saudi FDA. These are the status of the various therapeutic disciplines. You would see hematology oncology come on the top of the list. So we're not bad. It's not because of our active community, but it's because of the pharma industry. They're focusing more on hematology oncology. So there's more hem-oncology development worldwide than any other disciplines. And we get some share of this, so that's reflected also in our country. Hem-oncology first, endocrinology. You would see also benign hematology there. So if you combine benign hematology with malignant hematology, again, will become the first discipline. However, 40 or 44% of these clinical trials are observation. Again, they are not intervention trials. The companies will come to you and will collect data about multiple myeloma. Okay, give me your data about multiple myeloma. And they come to this hospital, they come to the next hospital and the other hospitals. And we go to our charts and collect using a data collection form and give it to the company. And what happened to this? Nothing. The company uses it as marketing their medications. They would see what's your practice, so they could target you and customize the practice to, again, bring awareness about their medications, etc. So they are not really impactful to the country. Yes, it may be helpful to some extent, but they are more helpful to the pharma industry to market their medications. So we're trying to, at our institution, minimize this and minimize the distraction of our physicians into these clinical trials, what they call clinical trials. And we try to ask the companies and impose on the companies to bring more intervention clinical trials, so to be part of either phase 3 or phase 2 clinical trials. And we incentivize the physicians. If he gets involved in phase 2 or phase 3 clinical trials, he gets more incentives than if he got enrolled in observation clinical trials. Again, these again pharma industry are our clients, and they are cooperative. They are actually they listen and they understand again the need to get enrolled in clinical trials. And again, here is the top pharma industry who are supporting and bringing clinical trials to the region. AstraZeneca on the top of the list. Okay, these data from KFMRC. As of recent, we have 147 clinical trials ongoing at KFMRC. 96 of those are intervention clinical trials, which is not a bad number. And the rest are non-intervention clinical trials. So again, we're trying to enhance this at our level. And here again, from our data, we have a big jump actually in 2024, and hopefully we'll see the same in 2025. And here is again the pharmaceutical industry. AstraZeneca again are most supported, Novartis and Obi, and we present this slide to the pharma industry, and we show them what's their status and what's their contribution again to trials in the country, and we see a response. We get actually on an almost weekly basis, we get a global team. It's not because of us, because now the country is pushing towards clinical trials, and I'm sure you've seen these visitors and global decision makers from pharma industry who are visiting the country, trying to enhance again the clinical trials in our region. Again, from KMR data, oncology is in the top of the list, ICU and cardiology. And again, we show this to our departments and we try to put them in competition and to see where they stand in terms of again, their contribution in clinical trials. All right, so these are the data, and now I'll talk about what's happening at the countrywide and what's the prospect and trends forward. This is a busy slide, but it shows how busy the country is in terms of innovation, clinical trials, research in general, biotechnology development, etc. So lots of institutions. You could classify pharma, biotech companies, research academia, innovation hub and accelerator, regulatory funding and investment, government support, etc. New entities that developed the RDIA, the Saudi NIH, the National Biotech Committee, Riyadh Biocentral, and you're aware of the recent launch of the Saudi National Biotech Strategy, and what's the importance of this, and how is it going to help the clinical trials? The next slide explains this. Now, if we are talking again biotech development. Now, any intervention, any biotechnology development will have to be taken through clinical trials to assess the success. So eventually, all of these developments will come through this path of phase 1, phase 2, phase 3 clinical trials. So our institution will get involved in this for sure. Now, this biotech development not necessarily developed here, and which we hope it would happen, but also when it's important from outside. And we see lots of, again, companies and small companies that are starting to, import these technologies, and they're looking for hospitals, again, to subject their product to assessment. So, how do we attract more clinical trials? And 4 domains that needs to, or 4 elements that we need to focus on: the speed, and that's again has been a hindrance or hindering factor. The speed of the process of processing approval of the clinical trials. So when you get approached by pharma industry to be part of a clinical trial, you need to go and seek for IRB approval, Saudi FDA approval, hospital approvals, whatever approvals. And these sometimes take months. I'm talking like 6 months or sometimes longer. The companies usually will not wait that long. They are investing. They have a product that they need to test, and they need to take it to the market as soon as possible. They have employees who are hired again for the development of this product. So they will, the longer the time, the bigger the loss they have. So they have to speed up their product development as soon as possible. On average, worldwide, the time taken to this is between 1 to 2 months for the approval process. Unfortunately, in our country, it takes up to 6 months to get approval of these. So that, and once you pay, once a company, one of the pharma industry come to you and with a clinical trial, and they will see your institution taking 6 months, they will not come back again. You lost them for probably for a number of years until their staff changes and their reputation changes or whatever. So it's very critical, again, to show the speed and show them that we are, we have the agility and we have the attention to speed the process. Let's say you get the approval in 2 months. Again, and most of the time you are given 1 or 2 or 3 patients or 5 patients to recruit, because it's a multi-center clinical trial worldwide. And each center will recruit this few patients. And if you happen to fail to recruit, and it's very common, what's the percentage of failure to recruit worldwide? Way higher, very, very shocking. It's 80%. 80% failure to recruit. And we always expect, when you see, lymphoma, for example. Okay, yeah, I have 100 patient lymphoma. You go to those lymphoma patients, you will fail to recruit one of them. And the reason is, there's the inclusion criteria and the exclusion criteria, which is usually strict. These clinical trials, they only select very specific and narrow indication. When it comes to reality, it's totally different. You will not be able to meet those narrow indications. And if you do, again, you come to, the approval of the patient, its acceptance to be enrolled, etc. So the failure is very high. And that's why these companies recruit in multiple centers worldwide. And they will come to you if they see you as a recruiter. And one of the reasons, again, for failure to recruit is we don't go and screen our patients. We wait for the patient to come to his clinic, and his appointment maybe after 3 months. And then you wait, every clinic, you just screen 2 or 3 patients. You don't have that data base that you could go to this 100 patients and see who is fit for the clinical trial. And this is common practice, unfortunately, in our hospitals. We don't have this database or registry that enable us, again, to identify those patients and call him to come. Oh, I have a clinical trial for you. If you want to participate in this clinical trial, etc. So this is common, again, reason for failure to recruit. Capable sites and reliable data, I don't need to emphasize on this, but I think this most of our setup is after this. The reliable data needs definitely training of staff, research coordinators, research associates, etc. They need to be trained again to the lots of documentation that's required for the clinical trials. And that's sometimes many physicians really, once they do one clinical trial, they will stop doing the other one because there's tremendous work that's needed. Okay, so now I will introduce you to the Saudi Clinical Trial Enterprise, and this is the website if you want to browse it. It's an initiative that we get from the NIDR, which is the National Industrial Development and Logistic Support Program, one of the major initiatives part of the Saudi Vision 2030. And it's to support again the pharma industry in the country, basically, and with it, clinical trials. We invited the stakeholders, the regulators, hospitals, pharma industry to be part of this. Again, to bring all of these stakeholders together to discuss the challenges, to discuss where we need to move forward, what's needed, what's many things. And again, this is open for all hospitals, for all institutions, And I'm offering a fight again to keep that medical system to be part of this. So the mission is to contribute to advancing clinical trials ecosystem in the kingdom, develop clinical research talents, enhance number and quality of clinical trials in the region, and ensure inclusion of the Saudi population, again in the population diversity covered by the clinical trials. Again, these are the objectives: establish network and multi-sites coordination, establish effective strategic partnership, promote medical research, address challenges related to clinical trial practice, and support economic diversification. All right, so what's needed again, and what are the elements? What sponsor wants to see to attract clinical trials? So we need to satisfy the sponsors of these clinical trials and show them that we are able to do clinical trials and we are able to satisfy their needs. Now, I listed a number of issues, and I highlighted some just to discuss. Of course, yes, we need high, standard of care. Most of these are available in our institutions. However, for example, higher recruitment rates, and I discussed this already. And again, we failed in many clinical trials. We failed in many clinical trials, again, recruiting patients, again, because of the issues I mentioned. Unable to spot or identify the needed, the fit patients as immediately as possible. And before that, when clinical trials, when a sponsor comes and approaches the institution, usually they come to the clinician. And when they come to the clinician, they will give the clinician a feasibility questionnaire. And the feasibility questionnaire, it's questions to the physician, asking them how many patients you have. Do you have this kind of patient? Do you have a patient with, for example, a GFR not less than 30, etc.? So the physician needs to answer this question. And then the company will decide to accept this physician on this site or not. Commonly, the failure is at this stage. It's not because the answers do not suit the clinical trial, because the physician takes ages to answer this question. And then, the company will close the site. So that's the first test for the site and by the company whether to include the site or not. So very important, again, message for our clinicians when you are approached by either the pharma or by your research institution to answer these questions in a matter of a day or hours or so. Because it gives an impression of how fast we're working. Well-functioning and predictable regulatory environment. Our Saudi day has improved a lot, and we see this actually. And they are now working again to improve with the Saudi NIH to improve the approval process. And they are aware of the importance of clinical trials, so they are pushing actually towards, again, accepting and attracting clinical trials. And that's a plus. Logistic complexity, for example, importation, exportation, etc. Again, we still have glitches here, and I think this will improve with the increased intensity of this. No unusual country requirements. Unfortunately, in the recent past, there are new entities that developed and asking these fermenters to again set the requirements. So that's again might hinder the process and the attraction of the trials to the country. And I think this is my last slide. The Saudi Group for Hematology Research. Again, this is an initiative, and I would probably suggest maybe that the two societies, again, the combined efforts, again, focus on enhancing clinical research and hematology. We need to talk to each other. Once we have a clinical trial in one institution, again, to open it in another institution, usually the pharma industry will appreciate this. When there are multiple sites open in the country for clinical trials, there's the insurance that recruitment is happening faster. More patients are enrolled. You recruit the 3 educated patients, you could ask for another 3 and another 3. It depends on, again, the speed of improvements and efforts. The group not only to conduct research, but also to discuss challenges and to discuss where we stand and how we develop plans and how we overcome these challenges. And I think it's very important, again, for us to, in order to move forward, to have these gathering under societies and under our research institutions. With that, again, thank you very much, and I hope that I added, again, some information and shed the light on the importance of clinical trials. Thank you. Thank you for the enriching talk. I'm sure everyone has questions. See Dr. Osman and Dr. Anwar writing questions, but wait for the discussion afterward. Now, Dr. Salim will introduce the next speaker. Thank you, Prof. Ahmed.
Thank you, Prof. Ahmed Al-Askar, for this excellent presentation. So we'll have the question at the end of the session. We move to our next speaker. It's my pleasure to introduce our next speaker, Dr. Maha Badawi. Dr. Maha is a consultant, adult hematology and transfusion medicine at King Abdulaziz University Hospital, associate professor at King Abdulaziz University. She is the director of the blood transfusion service, the director of the regional International Society of Blood Transfusion. Dr. Maha will talk about RBC phenotyping and genotyping in hemoglobin.
Thank you very much, Dr. Salim. And first of all, I would like to thank the organizers, everybody who helped organize this event and for the invitation. Very proud to be among you today. And Prof. Al-Askar, this was a very difficult talk to follow, but I'll try. So my topic is a little smaller. It describes our experience in our hospital with providing a better blood for our hemoglobin patients. Okay, so I don't have any conflicts of interest. However, as Dr. Salim mentioned, I am currently representing the Eastern Mediterranean region with the International Society of Blood Transfusion. The reason I'm mentioning this is please reach out if you would like to participate in the ISBT activities, if you have issues that you face in your workplace, research interests, anything actually that you're interested in achieving with help of the ISBT, please reach out. Everybody knows that at this current time, alhamdulillah, transfusion safety is significantly better than it used to be. The risks of infectious diseases transmissions is at a minimum right now. Alhamdulillah, in Saudi Arabia, the risk of acquiring HIV or hepatitis B and C is very, very minimal, comparable to Western countries because we follow the same screening tests and the best technologies. However, we still see other transfusion complications, including TACO, febrile reactions, and alloimmunization. Alloimmunization is actually common. The main reason for alloimmunization is the difference between the antigens of the patient and the donor. So antigen mismatch, exposure of the patient to antigens that the patient doesn't have through transfused blood usually results in antibody formation, so alloimmunization. The rate is variable. Not all antigens are immunogenic in a similar way. Obviously, the most immunogenic one is the D antigen, and we routinely try to match for that. However, there are other variables that contribute to the risk of immunization, including our genetic diversity and the inflammatory state of the patient. Immunization is a significant complication in patients who receive lots of transfusions. In studies published from various regions of the world, including the Middle East, the rate is up to 60% in some studies. And in our country, in Saudi Arabia, Dr. Walid Bawazir recently published meta-analysis. There were 14 studies that described the rate of immunization in patients with sickle cell disease and thalassemia, and he found that the rate was about 18%. Why do we worry about alloimmunization? Alloimmunization carries significant consequences to patients. So just describing the natural history of alloimmunization. So when patients are initially exposed to antigens that they do not have, if the patient was destined to develop an antibody, then we would call this the primary response. Typically, many antibodies disappear afterwards. The titer goes down to a minimum, what we call evanescence. With another exposure, the antibody titer rises again, causing delayed hemolytic transfusion reactions, and sometimes those are fatal. Antibody evanescence is seen in sickle cell disease up to 80% of the time. So that's a significant risk. So having antibody screening performed just before a transfusion might not pick up those antibodies, putting patients at risk of delayed hemolytic transfusion reactions. Other reasons we worry about alloimmunization is because of hemolytic disease of the fetus and newborn. Our sickle cell disease and thalassemia female patients who develop alloimmunization secondary to transfusion are at risk of developing hemolytic disease of the fetus and newborn, affecting the fetus and impacting their ability to have healthy children. And as I mentioned, the risk of delayed hemolytic transfusion reactions is very significant. In the United States, for example, if you look at data of over 5 years, from 2017 to 2021, so 5 years worth of data, these are the reasons of fatality, transfusion-related fatalities. So those are the patients who passed away as secondary complications of transfusion. And as you can see in the United States, hemolytic transfusion reactions because of non-ABO antibodies was up to 25 patients. So 25 patients died in the United States, secondary to delayed hemolytic transfusion reactions. And some of these patients were patients with sickle cell disease. Unfortunately, we do not have published data from Saudi Arabia. And to prevent this, the American Society of Hematology guidelines describe very clearly that they suggest obtaining an extended red cell antigen profile by genotyping or by serology, rather than only restricting ourselves to the minimum, which is ABO and RH typing. And if you look at the last point, here says genotyping is preferred over serological phenotyping as it provides additional antigen information and provides increased accuracy for some antigens. The guidelines also recommend prophylactic red cell antigen matching for RH and KIT antigens over only ABO and RH. So there are 2 steps required. One of them is being aware of the patient antigen makeup by serology or by genotyping, and trying to match for at least RH and KIT as much as possible. So not exposing the patient to antigens that they do not have. Why does the ASH guidelines suggest genotyping over phenotyping? These are some of the advantages of genotyping. So basically, the interpretation is computerized, it's streamlined, we can achieve high throughput using genotyping technologies. It does not require special reagents. Some of the antigens do not have anti-sera, so performing serological phenotyping sometimes is not possible. One big advantage is being able to know the antigen makeup of transfused patients, recently transfused patients, we would face difficulties trying to phenotype these individuals in the serological manner. So genotyping is extremely helpful for these patients, such as patients with hemoglobin. Genotyping actually can be applied in many settings. So in the patient setting, as I am describing right now, it's very helpful to phenotype patients who were recently transfused, patients with multiple antigens and high prevalence antigens, patients who have a positive DAT. It also has applications on the donor side, so you can phenotype many donors using genotyping technologies using this technique. So now I'm going to go to my experience here in working at Aziz University Hospital Blood Bank. The hospital currently now treats 150 patients with sickle cell disease and thalassemia through regular blood transfusion. 62 patients of these are sickle cell patients and 81 are patients with thalassemia, almost 150. 72 of these patients have positive antibody screening. It seems high, but I remind you, do not compare this to the rates I mentioned earlier. The rates I mentioned earlier encompass all sickle cell patients who are not, even some who were never transfused. But here I'm talking about patients who receive transfusions every 3 to 4 weeks. Although we try to phenotype match the red cells we give to our patients, this is not always possible because patients come to our attention when the antibodies have already been formed. Fortunately, the connection between the clinical side and the blood bank side is not very strong, so we're not always aware of the diagnosis. It is very rare that a clinician is ordering phenotyping before the transfusion journey started. So most of these patients, when they come to our attention, have already started transfusions, so phenotyping would become very complex. So a wonderful solution was presented by the agreement that we signed with the regional lab, the regional blood bank in the first health cluster for genotyping of patients and donors. And we have Dr. Nada here, the director there, which I'm very grateful for helping this come to life. So we already performed over the past few months, we performed genotyping for 48 patients, so about 30% of our patient population. That we are starting with right now. All results are reviewed by our physicians and are compared with what we know about the patient's phenotype. And because we already phenotype donors serologically routinely in an automated manner, then we can provide matched units once we know the patient phenotype, even if we do not have antibodies formed yet, so as a preventive among those 48 patients, we already have some very interesting results. So we already identified 6 patients with weak or partial antigen expressions, so little C, BD, and FEB. And there is also one case of a discrepancy affecting the E antigen. So the patient was initially thought to be positive for the E antigen and phenotyping, but phenotyping revealed that they were negative. And a probable explanation for this is because of phenotyping might have been performed after a transfusion, leading to inaccurate results.
5 minutes.
If you would like to read more about causes of discrepancies between red cell phenotyping and genotyping, this is a wonderful publication that I encourage going over. So we're very happy with this plan. Our plan is to try, with collaboration from the regional blood bank, is to genotype all patients with hemoglobinopathies, not only the transfusion-dependent ones. It would be wonderful if we can, on a national level, collaborate with other organizations to allow national access to patient data, including phenotyping, genotyping, and antibodies identified for every patient. And eventually, inshallah, with AI-enhanced technologies, we might be able to enhance and fasten the matching process by matching phenotypes and genotypes of donors and patients rather than doing it on a patient-by-patient basis, which takes a long time. What the future holds with the transfusion is we are on the way to develop universal blood in terms of ABO antigens. So researchers in Sweden and Canada already worked on transforming B red cells to O red cells by using enzymes and leaving the B antigen. However, there isn't much happening in regards to getting rid of the other antigens, so we still have to work on this to help our patients. And with that, I would like to thank Dr. Nadam Izdagar again and her staff in the regional blood bank for making this happen. Without the help of my team in the blood bank, Dr. Tariq Matwalli reviewing all the results, Mr. Mayman, the supervisor, Ms. Shahid Al-Johani developing wonderful electronic solutions to document our results, and also Mr. Majid Al-Zahrani, the hematology lab supervisor, for helping acquire those patients. This is my email and my X handle. If you would like to reach out in regards to this or ISBT activities, I'm happy to hear from you. Thank you so much.
Thank you very much for the excellent presentation. So, Ahmed will present the next speaker. My pleasure to introduce my teacher, Professor Saad Daouni. She is a well-known hematologist, oncologist, pediatric and adult as well. She is the head and founder of the prophetic medicine research chair of Cardiology Jameel, and she is a well-known researcher, and she will talk about a very important topic today about the nutrition and the impact on patients with hematologic diseases. Thank you, Prof.
Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you so much for kind presentation. شكرا لكيك فهد ريسيرش سنتر. هذا زمان احنا كنا من المؤسسين فيه. رائع والتغييرات ممتازة. اليوم I will go for the journey how the importance of nutrition in for precision medicine, and this is our goal in all our subspecialties. Just to go overall, what we mean the potential of precision medicine, just I choose pediatric hematology oncology, and how the importance of nutrition to improve the survival and decrease the side effect. And would like to see to focus about like, as you know, as oncologists, hematologists, you don't like our morbidity, which is the infection. Can you solve the resistance of infectious clone? And then we will go to conclusion recommendation. And as usual, I ask who's the first to start evidence-based medicine. Like precision, it's wellness, which is very important. It's not enough just to give medications, but you'd like to reach a normal, happy life for your patient and the outcome. And you would like the quality. Is this your patient satisfied or not? So the safety, very important. The first important, a proper diagnosis. Without proper diagnosis, you are not a clinician. How you treat without a proper diagnosis? As you know, pediatric oncology is a curable disease, but unfortunately, there is a lot of side, acute and chronic complications. So our aim of our study, like to see if nutrition can minimize these complications, the acute and chronic, which is very important for precision medicine. Proper diagnosis. You see a lot of researches. You are lucky to work in tertiary care medical center because without proper diagnosis, you are not reaching your goal. And if you see here, this is our unit of pediatric hematology oncology, which is established in 1990, which is a long time. But if you see here, a lot of departments supporting our patients. We see the same, a lot of departments in the diagnosis, a lot of the departments for supporting our patients. In our medical center, we treat all cases of pediatric hematology oncology. We never defer any patient. So, like we have a long journey of success. Even the lucky enough, we treat all patients, resident and referred to our center. So it's true, it's our kingdom, it's the kingdom of the humanity. And nothing to disclose. Okay, I will go with you with the journey of nutrition, and there is importance of nutrition in this unit. As we know, to see if the nutrition is very important and all its evidence-based medicine. There is two studies, unfortunate, very strange studies, one 1999, to show that if you give your patient antioxidant, what will happen to your patient? There is like interaction for your therapy. Other study 2015, it's the similar. If you give antioxidant, you will give your patient like the tumor will increase. Is this true? We'll see in our research. The first research, it's randomized clinical trial in this center in 2004 to see if this idea is true or not. And there is 8 departments associated. So our objective is to see the efficacy and the safety. To add, like it was Saudi honey. Most of our research is about our national product. This is the eligibility. Okay, all our clinical trials we presented in international meetings. So this study has been presented in European Critical Care of Hematology Oncology in 2012, and we published it. What we found, the group who had adding honey, Saudi honey, a lot of improvement. So if we see the mucous membrane, which is the first layer of immunity, we found markedly down with destruction of the mucous membrane. And what we found is very important, markedly decrease in infections of bacterial and fungal. So it's safe to add honey to your group of patients, which is the randomized clinical trial. And also, it's very important to get decreasing fungal and bacterial. And all the time when I go to any centers or any important, the major problem they have the fungal infection, and which we didn't have. Okay, let's go to the journey of research in honey. I'll try to be for 15 minutes. We love researches. We have more than 500 publications in our center. Okay, what we found worldwide, there is 1900 in honey. And we have one of the researches, it's very important to treat a peptic ulcer because the mechanism of anti-inflammatory, antibacterial. This is just, I would like you to read it because this is our publications to see the mechanism why antioxidant, anti-inflammatory heal the peptic ulcer and decrease the stress oxidative damage in two type of Saudi honey. Okay. Now we'll go to more problems. There is a start to be more problems, which is antimicrobial resistance. Now, because of a lot of using of antibiotic, start to have resistance to antibiotic, and let's see if honey can solve the problem. This is one of very nice, good article to show how in 5 type of honeys we tested like against the Staph aureus. And this is another one which is very important about Pseudomonas. Okay, even at Shigella, which is the common presentation of gastroenteritis, like honey have a good job. Okay, all the time we said in 2004 randomized clinical trial and in pediatric oncology honey, if there is a similar global international studies, yes, there is 5 international studies, but all in adult patients. So we are the first to start in pediatric and followed by a lot of adult in head and neck oncology and radiotherapy centers. Okay, we'll go for journey for another nutrition, which is the date, which you know date it's have a lot of important nutritional, but there is any medicinal effect. This is a study shows has been published in Molecules. We studied 12 types of Saudi date, and we found 42 benefit. Only 42. And more we see. I will not go the benefit with micro, macro, and all the important of it. The first study was published in our chair about the resistant Pseudomonas and resistant to imipenem drug, and it was a good study. All our clinical data, this is a second clinical data done in our center, and we presented internationally. Okay, has been published, okay, just to see the safety to add one type of date called Ajwa from Medina, and to see if this Ajwa can decrease infection or improve survival. Okay, design of the study, okay, exclusion criteria. So, as you know, unfortunately, our ethical committee gave us a long time, even this is nutrition. What we found, it's wonderful. We found in improvement in survival. This study, we follow our patients more than 10 years. Okay, even the fungal, bacterial, or marked without statistical significance. See the difference, even the hospitalization for febrile neutropenia. Okay, what we found more important, who had like cardiac problem, like usual, but the first case, it's a child Down syndrome with 4 medications cardiac, and she diagnosed at 3 years of age, and as this patient have big PSD and 4 multifailure, the other one, after the second dose of acute myeloid leukemia, he had cardiac problem. All of them, they are normal survive. Kaplan-Meier survival care for our patient markedly down for our patients who are on Ajwa as supportive care. Other things, what we found, like the educational, all our patients, while they are in chemo, they are having exam and having their tests. What we found, if you look to that patient, while he is on our ward giving chemotherapy, while he is in Ajwa, he graduated from high school 100%. Unfortunately, he is not Saudi, can we take him in medical school? Okay, so the conclusion, it's safe, it's very important, improve survival, decrease infections, and protection of the heart, what we want more. And now we go for nanotechnology and like patents. What we found here, I hope in the future will have support to have all our patents, UK, American patent to have start medications to prevent the side effect of chemotherapy on the heart and prevent the ischemic heart disease. This is the study. Okay, this is the patent, okay? And this is another study to prove that antioxidant has no effect on chemotherapy and it's safe. Okay, I will give you three about the benefit of treatment, not the treatment, how to prevent the tissue and how to prevent, cancer therapy, chemotherapy, radiotherapy, destruction. Like like if you take ajwa or treat, even date, it's very prevention. Okay, this is another study all in high impact journal, how it is anti-cancer, antioxidant, protection of liver, protection of kidneys, nervous system, and carry on. This is another study, so there is a lot of study we did about how the protection of tissue. And this one, now we need to have an over finding for a future direction for food and drug discovery. Okay, so the home messages, you need to have a good nutrition for all in health and in diseases for your patient, for the important, how the important of like research, food is very important for wellness. This is our survival. I would like to acknowledge all the departments supporting our unit, which is from diagnostic to treatment and also the research. This is a very important message. Please care about your patient. Wellness precision, it's you need to have a normal life, not only to give them medications. Who was the first start evidence-based medicine? We are the one. Ravi in 1865, he started evidence-based medicine, and we follow. If you look here, Dr. David Avey in 1987. It's too late, okay? I hope we continue for research for better life. Thank you so much.
Thank you so much, Professor Art, for the excellent talk. Now, Salim will introduce the next speaker.
Thank you, Professor Art, for your talk. It's my pleasure to introduce our next speaker, Dr. Aisha Ghaibi. She's an associate professor at the Faculty of Applied Medical Sciences, King Abdulaziz University. She established the pre-implementation genetic testing unit at King Abdulaziz University in 2014, and currently she is acting as the lab manager at the Institute of Genomic Medicine Science. Dr. Aisha will talk about pre-implementation genetic testing in sickle cell and thalassemia patients.
Assalamualaikum, everybody. Actually, I was planning to say thank you for giving me the applause, but we are in King Abdulaziz University, مركز الملك فهد للبحوث الطبية في المملكة العربية السعودية, I'm going to say it in Arabic. أشكركم على الفرصة الرائعة هذه في أنكم شرفتوني وأعطيتوني أن أنا أكون واحدة من السبيكرز وإني أعطي توك عن إنجازاتنا داخل بلدنا وفي أول مؤتمر لمركز الملك فهد اللي ناجح الحمد لله وإن شاء الله حيكون الجاي أحسن وأحسن. Now I'll switch to English. I'm going to talk about using the pre-implantation genetic testing in helping the patient of the thalassemia and sickle cell anemia. We all know that we have a high frequency of consequence marriage, and it's related to the transferring of the thalassemia and sickle cell anemia, which the government have decided to add it as the prenatal screening programs to help in reducing the occurrence of this disorder. Unfortunately, we all know that the people, after knowing that they are carriers, they still continue to demand. And some of them have already get married before the start of the application. So what the solutions for them, how we can help them? One of the solutions is the pre-implantation genetic testing. It's offered to the couples who have a high increased risk of having an abnormal baby due to the family history of genetic abnormality. And the PGT combines two areas of science. The IVF, it's needed as it's the source of the embryos. PGT offers different tests based on the abnormality that we are looking for. PGT-A, it's for aneuploidy screening, so abnormality related just to the number of the chromosome. PGT-SR, it's for structural rearrangements. Again, it's related just to the abnormality at the level of the chromosome, but structures, like translocation, for example. PGT-M, it's related to the monogenic disorders, means of the gene level, single mutation at the gene level. As I have said, we need for PGT before we need to go for IVF to increase the numbers of the available embryos to increase the chance of having a normal baby. So after they're doing the IVF, we go at the level of the embryos, either day 3 or day 5, and we do what we call it embryo biopsy. To take one single cell, as you can see in videos, if at day 3, or we do take 6 to 8 cells if we do it on the blastocyst stage. After that, to decide which test we use based on the abnormalities that we are looking for. So either PCR for single gene or HLA typing, array, next generation sequencing, or FISH, depends what I'm gonna look for. I will decide which technique we will use. For the thalassemia and sickle cell anemia, it's a single gene disorder. It's a mutation of the HPV gene. So the method that we usually use is the PCR together with the Sanger sequencing. And because there is HLA typing as well, so there are 3 different ways to decide how to deal with the family. So either we do direct mutation detection, or we do what we call it linkage analysis using the STR markers. Sometimes we use both. That depends on the case that we are dealing with. And I'll give them examples for each one. And what is the linkage analysis? The linkage analysis, which is the STR markers, here the microsatellite markers that we use on the same chromosome around the gene. And it's combined with something called short tandem repeat. As you can see, here, we have what we call a short tandem repeat, either tri or triplet CA, PG, PG, and this used for the DNA fingerprinting to know exactly what did the baby take from the mother and what the baby did take from the father. Thank you.
Sure. بوينتر هنا بس هنا ما اشتغل هنا
So you can resume for the time. Sorry for the technical issues. We'll have the discussion part until we have the technical issues solved. So we invite the speakers, Prof. Ahmed Al-Askar, Prof. Suad, Dr. Maha, for the discussion session. So any questions from the audience for our speaker? Mike. Magic.
My question to Prof. Ahmed. Prof. Ahmed, thank you very much first for your data and talk and interpretation. Biotechnology increase the drug discovery and innovation last 25 years since the human genome is finished, and we have also, revolution in Saudi Arabia by led by His Excellency, the Prime Minister سمو الأمير ولي العهد حفظه الله ورعاه for Saudi human genome. What do you think, in the future, 2030 or 2040, could be Saudi Arabia, since you are already in the industry of drug production, past the red line to the green line with developed countries, and even the cost after biotechnology innovation and discovery and technology from lab past stage 1, 2, 3, 4, and clinical trial to the shelf of pharmacies become $2 billion for one drug instead of $1 billion 50 years ago. Would you think we could come in even the expenses or the cost is very high now?
Thank you for the question. We understand the question, are we going to make it? And the answer is yes, we will. Now, how? Well, all the elements now are put together again to make it to the success. And again, to turn around from a consumer to a participant in research and development. We have, again, all these entities that was established, the research, RDIA, Research Development Innovation Authority, and Riyadh Biocentral, the National Biotech Committee, which is led by His Royal Highness the Crown Prince. Again, that gives you just an idea of how important how important this sector is to the Vision 2030. Now, developing one drug would be enough to lift us. I mean, the whole country. One drug itself, if it's developed or co-developed, not necessarily developed purely in the kingdom. By the way, no one individual, no one institution, no one country develop one drug alone. Usually it's multinational. If you take vaccine, for example, COVID-19, where everybody is aware, Pfizer vaccine, AstraZeneca vaccine, they're not developed by Pfizer. They're not developed by AstraZeneca. They're developed by a German innovator, and then partner with Pfizer, and that's how the evolution of and the sequence of biotech developed. So could Saudi Arabia be part of the sequence? Yes. It just, we're navigating the way, and I think that will happen hopefully soon. And again, developing one medications like Pfizer vaccine will give you revenue as the magnitude of the budget of the Saudi Arabia. Yes.
Question.
Assalamu alaikum. Thank you to all of our speakers. My question is to Dr. Ahmed Askar. Your talk was really comprehensive. Thank you so much for that comprehensive overview about the clinical trials. My question here is, what are the investigations that are mandated and required before initiating a clinical trial, please?
If I understand the question clearly,
Yes.
you are talking about the preclinical
investigations.
Oh yes, it's a long process. It's a very long process. It starts with the basic science. Now, and I always, by the way, encourage our people not to spend time on basic science. It doesn't mean that it's not important. It's extremely important. However, there's abundance, I would say, of basic science already there, and just focus on the translational research. That means use the basic science that's already developed, scan it, whatever your field is, and see how you take that data to help you again into animal testing. And to, for example, you already discovered the gene, and that gene is already, there are people working with that gene mutation to target it by medication. There could be a medication that's already developed. Why didn't you think of another medication to target? So that pathway is successful. Why did you target it also? Inhibit it by another molecule, or use another medication, repurpose it. So another medication that's already tested, why didn't you use that medication for another medication and just take it to phase 2, phase 3? So there is lots of things to be done. Not necessarily to start from A, or the basic, the very basic. Try to explore what new genes. We don't need to identify new genes. There are genes already identified. There are pathways already identified in the diseases. Just target those pathways with molecules and medications, and either new molecule, or there is a molecules that's identified and already tested, and just repurpose it to target that pathway. That's the translational. Take it to animal testing, and then you'll be ready for phase 1, phase
2. Great talk, Dr. Ahmed and everyone else. So my question is about the preclinical models, because I think they are important. Do we have
through these specific patient-derived xenografts or patient-derived organoids to distribute drugs on, just to make it population specific before it goes to clinical trial.
Do we have organoids developed and tissues
Taken from Saudi patients, like xenografts or organoid models, because all the models that are available, like from other populations, like Caucasian models, and we need to test it on our population, right?
True. I'm not aware of organoids developed and tissue screening from Saudi population. But I mean, the floor is open. I mean, if you're capable, why not?
Doesn't have to be organoids, also xenografts, yes, xenografts in patients.
Any other questions from the audience?
Well, I have too many questions. I don't know. It should start. First of all, I would like to thank everybody for the great talks. I have a question to Dr. Aisha first. What's the success rate in PGT in thalassemia sickle cell?
You mean the success rate of the pregnancy?
Yeah, well, yes, pregnancy without the disease.
Yeah. In case of without the disease, I'm going to say that the rate of the success of having the normal baby, not having that abnormality, is 100%. Because we don't do transfer any baby that we are even if we have just 0.111% of the result. We only transfer the baby that is completely normal for that only mutation, not for any other mutation. For the pregnancy, it depends on the situation of the mother. The hormonal stimulation, the age of the mother, the process of transferring the embryos. If it is fresh, the pregnancy rate is going to be a bit low because still she's under the hormonal stimulation. If they delay the embryo transfer after 4 to 3 months and freeze the embryos, the rate of pregnancy becomes higher because it's going to be done in her normal time of the period.
Can I continue? صراحة it's very important talk, Prof. Ahmed. Today, I think it's a national mission to work together to have to maximize the benefits on the country and the region from clinical trials. Today, we're always talking about value-based pricing of medications and the benefits patients gain rather than just approval and prescribing medicine to everybody. And we have very unique population in Saudi, unique populations in Saudi and communities in Saudi and the region. And I think we should capitalize on that to attract pharma, not only to do clinical trials, to invest in training, to invest in education, to invest in building infrastructure. And this is part of the national goal of localization and contribution to the economy from these companies. And I think we have, through the SSCTE, that enterprise group, I think we could have working group that helps unifying us. The problem is we don't need, I think we have to address the elephant in the room everywhere. I think we try to compete. I think we should complement each other, not compete. Work together. It's a national goal, it's not a personal goal today. And we need to reach there and then we can compete once we're there. I think maybe we can have a group that could deliver our pain points, the obstacles we're facing. Try to complement, find the ways to complement each other as a whole nation to the stakeholders. FDA, we have an amazing FDA.
today our goal is to be number one in the world, right? So I think this is something I think about, and I think we could do it through the already established enterprise. And that would help also in the networking. Yesterday we had great talks about rare diseases and how from a single case, very single rare genetic defect, they ended up with 20, 30 cases because of the network. Your presence here and everybody's today, I think that could help to do that. These are just ideas I love to share with everybody. And I would love to hear you.
I can't emphasize more on what we said. Absolutely. I think we're supposed to work as one team, leverage the resources that's available in the country, establishing these groups again, and to have everybody again supportive of their mission. The SCTE again established to maximize the benefits from the pharma industry, benefits from the resources available. All what we need is just combine the efforts. Now, and understand what's needed. Again, there are challenges. What's needed to be done to overcome those challenges? There are strengths in this institution, strength in the other institution, strength in this individual, in the other individual, and that's how we could make it. As I said again earlier, and no one individual, no one institution, no one country could do it alone. So if we understand this and working together, I think that's the key solution. Discussion, communication. This gathering is again is one of the tools that could be utilized.
Thank you. I have one question to Dr. Omaha regarding the same line of collaborating with other partners in the country. Genotyping is very important, I think, and as you shown in a patient with sickle cell disease. And sickle cell disease is a big burden in the country. And what are the steps or is there an initiative that's going on to establish a reference lab or collaborative work on getting genotyping across the country?
Thank you, Dr. Ahmed. Actually, we will not be able to help those patients without collaboration, as you mentioned. And I think what I presented is just an illustration of collaboration with the services provided freely by the Ministry of Health was very helpful to patients. And so the Ministry of Health actually advertised and reached out to the hospitals, to Dr. Amanda, I mean, very, very helpful. She actually sought our patients, not for any benefit except for patient benefit. So I think that's a great initiative. And I know there are other centers in Saudi Arabia who are doing the same. Reference laboratory is something I know the Ministry of Health is working on. So we're very hopeful that we will see that come to light.
Any other questions? Okay.
Thank you, everybody. I would like to thank our speaker for their presentation, the audience for their questions. We conclude this session with this. We will have a 15-minute coffee break and we will resume our session. Dr. Aisha will resume her session that we stopped earlier.
بس الأول
ساعتك بنتشرف ان شاء الله. Come back, please.
That's all. It's like that.
Back to the lecture, as I have mentioned before, we use different ways for the analysis for the EGT, either direct mutation detection or using the linkage analysis or compound detection. And I have explained what is the SPR markers and the linkage analysis and how it's related to the DNA fingerprinting to know exactly what the baby gets from the mother and the father. Not sure. Okay, as you can see in the chromosome from the mother and the father, there is number 2 proteins and they are sharing. So to do the linkage analysis, there is an important things you need. You need to have a reference baby, either normal or affected, to be able to tell that this is the normal allele and this is the abnormal allele. The DNA fingerprinting, it just tells you that this comes from the mother and the father. But without the reference, you won't be able to tell which is the normal and affected. For example, this is a case, it's an autosomal dominant, so the mother already affected. and they had an abnormal baby, and through this linkage analysis, and this baby is our reference, we managed to know exactly what is the affected allele and what's the normal allele. And based on it, any baby that will have this abnormal allele won't be transferred while the others are okay. So I'm going to give an examples for the three different ways. This is a case of a family. Both can see the mother and the father are carrier for beta thalassemia, however, two different mutations. The father G to T and the mother G to A. Unfortunately, they came to us with two affected babies having compound heterozygous. It doesn't call homo, it's a compound heterozygous because the mutations they took from the mother and father are different. But they are on the same gene and on the same chromosome. Also, this family asked for doing the HLA matching because they want to have a baby that's matched with the boy and the girl to be able to use to do blood transfer, transfusions, or bone marrows. So we included the HLA. We all know that the HLA genes are located in a totally different chromosome, not on chromosome 11 where the HPV genes is located on the PR of chromosome 6, which is well known with a very high level of polymorphism. So we tried to cover the most area of the HLA, and then we started to do the haplotype. Which way we will use when we went through the family, because they were cousins. So as you can see, there were some match with the alleles. For example, 219, 219, 219 in the normal allele, of the affected allele, the mother with the father. So if we depended on the linkage analysis, this would cause misdiagnosis. So we decided to use the third way, which is compound analysis. The linkage analysis plus the mutation detection. So any baby will have these two alleles, like the affected baby will not be transferred, while the others are okay. When it comes to the HLA matching, what we found that both the girls and the boy, they took the same allele from the mother, which is the red. However, unfortunately for the alleles that they took from the father, the girls take the red while the boy took the green, which makes them unmatched. So this is going to make it a bit difficult to have one baby match for both of them. So the baby that will come either for the girl or for the boy. So we went for the IVF, and we managed to get only 4 embryos. All of them were normal, they are carrier, but unfortunately, one of them was matched with the boy only. So they have decided not to do any transfer now, to do more IVF to get more chance and more babies. The second case, it came to us having both. The father was carrier for the thalassemia and the mother for sickle cell anemia. Unfortunately, they thought that these two genetic disorders away from each other. They said we will go for normal pregnancy. But unfortunately, the first baby came to them was a girl affected having both thalassemia and sickle cell anemia. So they decided to do the PGT again together with the HLA matching. When we went through the family, the great things, they were not the cousins. So you can see that their alleles were totally different. 21, 49, 52, 39, 55. So the informativity is very high. So it was easy for us to get the affected allele. So any babies will have these two affected alleles won't be transferred, and while the others are the carrier of normal okay. And even for the HLA matching, it's just the one girl, it's clear what she took from the mother and from the father. So it's the opposite. In the mutation, we don't want the baby that's similar to the affected. While in the HLA, we want the baby that is similar to the affected. Out of the 11 embryos, 4 of them were affected, so we excluded them. And out of the 7 embryos that were normal, only 3 of them were matched with the girl. 2 of them were carrier exactly, and 1 of them was a completely normal boy, not even a carrier. So they have decided to transfer two, and alhamdulillah, she became pregnant, and she delivered a normal baby, and they went for the HLA, and the matching was over 85%. The last but not least, the case. A family, they found that they are also carrier for beta thalassemia for the same mutation exactly. So indeed, the first baby came to them was affected. But as you can see, a totally different gene, a totally different mutation away from the HPV. Years after, another baby came to them, a girl, also affected, but again, totally different third gene away from the HPV. So we told them, definitely you need to go and check for the family, because as you can see, both of them are homo, which means definitely the mother and the father will be carrier. It's not an autosomal dominant to say it's a de novo mutation just appeared like this. So they went for the screening, and the mother and the father were carriers for the two mutations in addition to the HPV. Again, it's a placebo experiment. So to decide which way we will do, which protocol, we decided to go for the single mutation detection, much easier, because the three mutations are in three different genes located in three different chromosomes. So if we're gonna go for the linkage analysis, this means over 17 or 19 primers, that will increase the mutation, the contamination, the allele dropout, so could become misdiagnosis. So what we just did the single mutation detection. As you can see, for example, embryo 7 having the 3 mutation and affected baby. So out of the 14, only 4 of them were normal. 3 of them were carrier exactly for the mother, like the mother and father for everything. 1 of them was carrier for just the HPV, but normal for the rest of the mutations. And they did go for the transfer, and I think she became pregnant. And they have contacted us, they want to do later another transfer for the other 2 babies. Finally, in conclusion, the PGT, like any technology, has advantage and limitation. I'm not going to say disadvantage. The advantages, you start the pregnancy knowing that the baby doesn't carry that specific genetic abnormalities. You prevent the genetic disorder from being transferred to the next generations. And really, some of the families, when they came to us, they said, we don't want even to transfer the carrier baby. Even a carrier, it is normal, but we don't want to transfer because we want to stop this disorder from being keep transferring within the generations. Finally, alternative to the prenatal diagnosis to avoid the termination of pregnancy because of the ethical issues in our country for the termination of pregnancy. The limitation, you have to go through the IVF, which is a very high cost. Without it, you can't do the PGT. Sometimes all the embryos not suitable for transfers. Thus, you cannot guarantee that the baby is not having any other abnormalities, for example, trisomy, translocations, or anything else. This the message from the conference, vaccine جودة الحياة, and that's what the main aim of the PGT. إن احنا نحسن جودة الحياة, improving the quality of life by preventing the transfer of the genetics abnormalities. Finally, we would like to thank the team, the PGT, the molecular records. And by the way, this is not a draw, this is an embryo. But unfortunately, it just stopped at the 2-cell stage and arrested with a smile.
Excellent talk despite the technical difficulties. Thank you again. And we'll probably proceed for the next session. 11? 5 minutes? Okay, 5 minutes and then we'll start the next. Thank you.
We will start our next session for today. First of all, I would like to apologize for the technical issues in the morning, and I hope that you will enjoy the rest of the day without any technical issues. Our moderator for this sessions, Dr. Abdullah Muhammadi. Dr. Abdullah is an assistant professor and consultant in hematology at King Abdulaziz University, the board certified in hematology and oncology by the American Board of Internal Medicine. The second moderator is Dr. Majid Al-Majari, an associate professor of hematology at the Faculty of Applied Medical Science and a consultant in clinical laboratory hematology. He earned his PhD in clinical laboratory sciences from the Catholic University of America and the NIH in 2020, and his research focuses on developing RBC biomarker for acquired and hereditary anemia and advancing personalized medicine. Thank you very much.
Morning, everyone, and welcome to today's session. It's my pleasure to introduce our first speaker, Dr. Rahman Radwi. Dr. Rahman Radwi, he is internal medicine, hematology, and stem cell transplant consultant. He's associated professor at King Abdulaziz University and principal investigator at Hematology Research Unit in King Fahd Medical Research Center. He will be presenting the important topics of pharmacogenomic in thrombosis. We are honored to have you here today, and the floor is yours.
Assalamu alaikum. Thank you so much, Dr. Abdullah, for the kind introduction and also for the organizing of this conference today. Today, I will be talking about pharmacogenomics approaches in thrombosis. Just to look at precision medicine, and then we keep going on with the same thought because this is actually what we would like to go with wellness behaviors for our patients. I would also like to tell you, just continuing with the theme for today morning, I'm also the clinical trials director at the university. So the mini professor here at the university, and we're trying to attract so many clinical trials as much as possible as we can, and this could not happen unless we have a unit where we accumulate all the experience, try to facilitate the conduction of these clinical trials in any field, not necessarily in hematology, but any field of medicine. Moving forward, Bismillah. Okay, so my objectives today are very short and up to the point. It's going to be introduction, trying to connect the dots, or trying to connect the dots together, not necessarily understanding each dot because it will take more than 15 minutes, but just to try to understand the role of pharmacogenomics in the current era. And we're going to try to bring some examples from both, like in pedigree warfarin. So the term pharmacogenomics, it's a wide term, but it has been shortened to go into specific area, which is how is the body going to interact with a certain drug based on a genomic variation that the body does have. it will entail the pharmacodynamics, it will entail the pharmacokinetics, but what it makes it different from the other. Now we have more than 2,000 genes that we can actually do tests, and if I went back to 2010, it was only 500 genes. So it's a highly growing field, and I actually managed to go into this website, the PharmGKB, which helped me a lot in this talk and presentation. I advise you to go and take a look at that. So again, why do we study pharmacogenomics? Definitely because we would like to know which individual responds to the drug two ways. Either it's efficacious and is it safe or not for my patient. Also to identify specific target areas of risk for the treatment, and then identify those at high risk or low risk of getting the benefit from this medication. I'm going to repeat this slide again at the end of the talk just to answer these questions. So pharmacogenomics, what we usually do test, we test the SNPs, single nucleotide polymorphism. We sequence these variants and get an actionable one. Those actions are actually different. Probably 2023, some SNPs were not actionable. And then with the growing field of additional data, additional research, in different population, like this works in the European, in the Japanese, and then we found, okay, it works right now in Saudis. So this is why we need more research in this area. And I would like to use an analogy by basically regarding the SNP. If you have, like, for example, a certain model of car, like let's say, for example, Toyota Camry, it's going to take you from point A to point B. It's going to efficiency with gas and environment. But the difference would happen if you have a different color. If you have a white color Camry or a black color Camry, the warmness that it gives, the attractiveness for certain colors, it's actually what makes it different. Why I mention this? Because we're not talking about mutation. We're talking about a normal variation in the population according to the single nucleotide polymorphism. So with precision medicine, you have a patient who comes to the hospital with pneumonia, you're going to give third-generation cephalosporin, macular, azithromycin. And this is what we call usually one size fits all. But unfortunately, some patients would not actually get the benefit and instead probably get harm. So the optimum medication is where you have the same diagnosis, you personalize your treatment according to which group is coming to my care. This is kind of a report that usually we get when we talk about pharmacogenomics. You send your test to different labs, and there are many labs in the world. Some labs are actually non-profit, and some labs would like to get it as a business model. So you get different reports. Remember, there are more than 2,000 genes that can be tested that can affect certain indications of working. And this is one related to our hematology, and we talk about hematology, we talk about the benign and also the malignant. So benefit to the patient. So it saves money on the long term, prevents side effects, would improve the quality of life, and as I mentioned, because you're looking at that precision medicine. What benefit it can bring to the institution? So it would decrease the adverse drug events, will lead sometimes to decrease the failure of those clinical trials that you would like to conduct. It will also decrease in time required for drug approval. When you face an insurance company, you'd like to say, now we have a study approving that this certain SNP actually, this expensive drug does work for them, so you get the approval faster. Also, the time the patient needs to be on medication, whether a year or 6 months, depends, because I gave him the most effective medication. Okay, shifting gear, giving some examples. I'd like to bring here a nice story about Plavix or clopidogrel. So this study was published back in 2003. Patient presents with MI, he goes to PCI, put a stent, drug-eluting stent, and then you have to keep him on antiplatelet aspirin. They used clopidogrel in the past. The first generation moved to second generation clopidogrel. And then they found it gives marvelous results, prevents instant thrombosis and recurrent MI. So, okay, patients need to be on aspirin clopidogrel. Later on, they found the patients on the far right side on Plavix, actually they need more, they're highly sensitive. And patients on the far left of the column chart, they don't get any benefit from Plavix. So what's going on? What's the problem? Looking into actually, Plavix is a prodrug. It needs to be metabolized by the liver with certain SNPs available in the body. Finding out the SNPs moving forward, okay, it's CYP2C19. So should I test my patient for CYP2C19 before I give him the clopidogrel? And because those ones with the dotted red line actually have reduced functionality, they are high risk of developing MI stroke. So if you gave it Plavix, they're not going to benefit from it. And this actually led to an FDA label suggesting that actually you do these tests, you advise your patient that probably he has this certain mutation, a certain SNP that he might not benefit from clopidogrel. This was back in 2010. At that time, you can understand, and most of us will know this, testing for genomic testing is actually going to be difficult. Even if you're going to do it, it might take weeks or probably sometimes up to a month till you get the result back. But I have a patient with MI who I need to give my medication right now. So that was a little bit of a problem. So they were thinking, okay, now I have a problem with this medication. We need to invent a new drug. So they came up with new drugs, ticagrelor, bisagrelor. So they tested in a New England Journal paper here, and you can see that, okay, whomever we found that he has this SNP, we're gonna give him ticagrelor, and they actually found benefit. They tried it even with MI, not only with TIA. Tried it with MI, they showed a marginal benefit. So not that very improvement when I switch patient from ticagrelor to clopidogrel. And also, they said, okay, whoever we found that he has this SNP, how about we double the dose of Plavix? So trying to give him the highest dose possible. And actually, as you can see on the far right side, it's actually as much as that the patient has the reduced allele function. So Plavix testing for that doesn't work. And why testing for that doesn't work right now? It's basically because not every patient coming with a cardiovascular disease we do genetic testing. Prospective trials failed to show that actually knowing that the patient has this SNP would add extreme value and benefit, and also some other competitors. So if you don't like that Plavix, try something else. The market is actually full of other medications that actually can provide with benefits for these patients. So moving to thrombosis, not necessarily my field, but I love it. So actually the thrombosis starts even from before the patient is born. If he actually has carried risk for developing thrombosis later on in his life, that's something that he would inherit. And it will, knowing this information will also affect the kind of treatment I would like to give. I'm not going to talk about the diagnosis or who patients who are actually going to get it. This is a field called thrombophilia, inherited thrombophilia, and the testing. So I'm not going to touch aspect on that, but we're going to talk about when we treat because it's pharmacology, because I'm giving a drug back. So starting with the warfarin, you have two types of warfarin, the S-warfarin, the R-warfarin, both are targeting to stop the reduction of the vitamin K. So through the B core C1. So targeting the B core C1, targeting the CYP2C9, and also the CYP4L2 are affecting the pharmacodynamics and sometimes the kinetics of the drug in the body. Knowing that information, there has been an international consensus guideline regarding pharmacogenomics in warfarin for particular. They published multiple algorithms, went into asking people, okay, if you found this genetic mutation for the patient, you're going to change the dosing you're giving for the patient. So how did that affect? So in terms of prevention, like atrial fibrillation on the left, or treatment for warfarin and for DVT, of course, always the problem why we want to affect the dose, because warfarin has something very common, it's called narrow therapeutic index. If you're on the right part of this chart, you're going to bleed. If you're on the far left of this chart, you're not going to get any benefit, and the window is too small. So you have to keep the INR, for example, between 2 to 3. So very hard, especially in cardiologists, to keep these drugs affecting INR. How about we just give 5 milligram of warfarin to everyone? One size fits all. Just give 5 milligram to everyone. That's the results. Lots of bleeding, lots of recurrence of cardiovascular. 5 minutes? Okay. So try showing warfarin dosing. They said, okay, how about we combine the pharmacogenomics with the warfarin dosing, and this actually led to FDA asking that you do the CYP2C9 B4C1 genotype when available, only to assist you in the starting dose. Because later on, you're just gonna change the dose, add 1 milligram, remove 1 milligram, and that's it. So this actually, even when they actually showed how about we combine clinical algorithm for pharmacogenomics, they found no difference. There's no difference. You just go ahead with the clinical algorithm, and it no need to test for VOC1 or the other mutations, the other SNPs for warfarin. So again, the question why genetic testing did not help in warfarin dosing, because again, we don't do the genetic testing for treatment, we do it for diagnosis while the patient develops, but we don't do it for testing. Their prospective studies did not show convincing benefit. And again, we have new drugs in the market, which are the DUAs, shown in the patient. So DUAs, not to be forgotten, I brought here 5 examples of DUAs, I only use 4 of them, all of them except for the Detrexaban. SNPs actually do affect in activation, transport, and metabolism of the drug. And we know they are efficacious, but how about the safety profile? Is everyone receiving apixaban and peroxaban, the digoxin will actually not bleed heavily? And they did it, there was a small study. Of course, we do have, there are no great studies in that matter because not in thrombosis, we extract DNA and try to test the SNP for each patient. And we try to couple whatever in the registry with the biobank. So actually found certain SNPs affecting the bleeding pattern. But those are very, very, like 200 patients, 300 patients, not like in cardiology when doing with the clopidogrel with like 10,000, 50,000 patients. Also, very recent, I apologize for the small picture here, but also looking at the left one, you can see that the blue lines are showing that if you're getting apixaban, A, which is left upper corner, if you're getting apixaban with a certain SNP, you're going to bleed less. The second graph just decided if you're getting rivaroxaban with a certain SNP, you're going to have less recurrent thrombotic events. And the data goes on and on. You just need to look up for these SNPs, try to correlate it clinically and see who actually is going to get the benefit. So are we all born the same? I try to bring it. So testing for SET1 or ABCD1 are the commonly one tested for DUAs, safety. And people did it for Caucasian and Russian data, and actually found it's actually different. So definitely, I would like to say that we actually need to do studies of our own population, knowing who's actually going to develop this or not. And if you look at the Saudi literature, it's actually cool, especially when you correlate pharmacogenomics to the efficacy of even warfarin or DUAs and other anticoagulation or even antiplatelets. But luckily, actually, a few months or 9 months ago, the King Faisal Hospital of Riyadh actually announced the launching of a special clinic in the hospital called pharmacogenomics, where actually you need personalized, they use personalized medicine. If you think your patient is not getting the actual benefit of this medication, or you suspect that they might not get the actual benefit, send them to this clinic and they actually going to do the testing that are actually well approved worldwide. So again, last slide of my talk is again to answer the same question that I did told you before, why we do test for pharmacogenomics. So when I said it modified the individual response to a drug, it's actually difficult. Why? Because no huge investment going into that area because new drugs are always coming. So why should I test? I need a better drug that has better safety profile, for instance. Does genetic identify a specific target mediator risk for treatment? I haven't touched on this session. But the last one I'd like to emphasize on is identify who at high risk of an outcome and may appreciate greater absolute benefit. That actually has done, because we as clinicians, like I said as a clinician, we prefer simple risk scores that I can do in my clinic, and then I need a quick answer. So I'm just going to rely on laboratory tests that are actually going to bring the results there. This is also giving me a chance to tell you that the rapid advances in the genomic area, where you can sometimes get the results of the test, either at a point of care or even spontaneously on the same day, would actually affect the way you're going to decide about which patient to take certain medications or not. The standard slide is actually dedicated to everyone who travels from Makkah to Jeddah every day, okay, to commute to work. So I have also a friend here telling me it's close to me. So تحية للعلم والعلماء. مكة. This is Makkah to Jeddah.
Thank you, Dr. Ahmad, for this excellent presentation. Now it's my pleasure to introduce our second speaker, Dr. Arij Ahmadi. Dr. Arij is Assistant Professor in Coagulation and Hemostasis at the Department of Medical Laboratory Science and Faculty of Applied Medical Science. She is currently affiliated with the Immunology Unit, where her work focuses on exploring antithrombotic and vasoprotective mechanisms under the hemodynamic regulation. Dr. Rich, we are honored to have you here today, and the floor is yours.
Thank you. Thank you for the introduction. Good morning, everyone. I'm very pleased. It's really an honor to be here today. Thank you for having me. So, this morning, I would like to take a closer look at how endothelial cells respond to shear stress, or how the hemodynamic forces influence the process of thrombus formation via the vascular endothelium. Let me first declare no conflict of interest. This talk should only take 15 minutes, so I'll briefly walk you through the effect of shear stress on the vasculature, some of the key shear responsive antithrombotic and vasoprotective molecules, my recent findings, and their implications. With that, let's dive into the session. Endothelial cells are the innermost layer of the blood vessel wall facing the flowing blood, and the hemodynamic forces, or the shear stress, is the blood flow force that act on the surface of the endothelium. Cells in the straight arteries exposed to unidirectional blood flow that has high magnitude of shear stress, while cells in veins exposed to disturbed blood flow that has a low magnitude of shear stress. So, as mentioned, endothelial cells who are exposed to different magnitude of shear stress has different phenotype. Those exposed to unidirectional high shear stress are aligned with the flow. Yeah. Unidirectional high shear stress influence the endothelial cell behavior. So high shear stress or unidirectional flow influence the alignment of the cells. So cells in their unidirectional flow are aligned with the flow. They are antithrombotic. They prevent platelet adhesion and aggregation. They are also anti-inflammatory. They have balanced redox system, and they maintain functional vascular tone. On the other hand, cells exposed to disturbed blood flow, they are misaligned, and they are prothrombotic. They are pro-inflammatory as well. They have a high oxidative stress, and they are permeable to macromolecules. So cells experiencing this condition, and with those phenotype, can develop arterial thrombosis and activate platelets. This can also develop chronic cardiovascular diseases like atherosclerosis, and it is well known that atherosclerosis is associated with myocardial infarction and stroke. So this always raised the question of what maintains functional endothelial cells under high shear stress conditions. Endothelial nitric oxide synthase, or ENOS, is a key protective vasoprotective molecule that is upregulated by high shear stress, and the glypican-1, which is a major component of the glycocalyx, since the shear stress stimulation and transduce this mechanical stimuli into intracellular activation of ENOS, and ENOS produces nitric oxide. Nitric oxide is antithrombotic. It inhibits platelet aggregation and adhesion and activation. It is also a well-known vasorelaxant when it interacts with the adjacent smooth muscle cells. It is also anti-inflammatory and vasoprotective as it provides as a protection from atherosclerosis. According to previous studies, mice lacked enos has higher level of platelet aggregation. So the question is, what regulates enos mRNA stability under high shear stress? Another vasoprotective molecule is prostacyclin synthase, and again, high shear stress stimulates the mechanosensor, GLP-1, and transduce this mechanical stimuli into intracellular upregulation of prostacyclin via the prostacyclin synthase. And the prostacyclin is also antithrombotic, vasorelaxant, and protect from atherosclerosis. It has been identified that a single point mutation in the prostacyclin synthase gene has been associated with myocardial infarction in Japanese people. Again, what regulates prostacyclin synthase mRNA stability hasn't yet been identified. In this talk, I'm focusing on the ankyrin repeats and the single KH domain, or ANKHD1. It was first identified in Drosophila as a JAK-STAT pathway regulator, and subsequently it has shown to be a Hippo pathway regulator. Therefore, it has been studied in cancer. It's involved in tissue growth. ANKHD1 has shown to be a regulator of clear cell renal cell carcinoma, while its function as a microRNA binding protein. It can also bind to a lamina coding RNA in glioma and endothelial cells to promote angiogenesis. It can co-localize with caprin-1 in stress granules under oxidative stress conditions. So the function of this interesting RNA binding protein in the normal vasculature and in non-cancerous cells hasn't yet been identified and is the focus of my work. First of all, I wanted to assess whether shear stress has any influence on ANKHD1 levels. So I assessed the effect of shear stress on ANKHD1 in human coronary artery endothelial cells and the human umbilical vein endothelial cells. High shear stress significantly upregulates ANKHD1 mRNA and at high shear, ANKHD1 mRNA and protein at high shear stress conditions in both cells. And also I assessed the levels of ANKHD1, particularly in endothelial cells in the primary arch. The primary arch has areas exposed to high shear stress in the outer curvature. And in the inner curvature, the primary arch experience low shear stress. So I can quantify the levels of ANKHD1 in different areas within the same mice. So ANKHD1 was significantly upregulated under high shear stress conditions in the outer curvatures areas protected from atherosclerosis and thrombosis. And this indicates that it perhaps has any function with these protected endothelial cells. So this always raises the question of what is the function of ANKHD1 in the high shear stress condition or in the protective conditions. So I silenced ANKHD1 via ANKHD1 specific siRNA in human coronary artery endothelial cells exposed to high shear stress and sent the sample for sequencing. And as you can see, fluid shear stress and atherosclerosis was one of the top enriched pathway. And this indicates the involvement of ANKHD1 in shear stress pathway under the sensation of hemodynamic forces. And one of these pathways is enos and prostacyclin synthase. So I first assessed the effect of shear stress on enos, and this comes in agreement with previously published work. Enos mRNA was significantly upregulated at high shear stress conditions in human coronary artery endothelial cells and in HUVEC or human umbilical vein endothelial cells. And this also this confirms previously published work and also confirmed that my experiment works. So the high shear stress influenced the function of the endothelium and upregulated the well-known high shear stress marker. While a study has revealed a shear stress responsive sequence in the prostacyclin synthase gene, whether prostacyclin synthase is shear responsive, this hasn't been experimentally tested. So I found for the first time that prostacyclin synthase is significantly upregulated under the response of high shear stress compared to the low shear stress at mRNA levels and also at protein levels in different primary human endothelial cells. Now, whether NKHD1 regulates enos and prostacyclin synthase, I inhibited NKHD1 using different siRNAs, and it shows that NKHD1 attenuated the effect of shear stress on enos and prostacyclin synthase, indicating the positive regulation of enos and prostacyclin synthase. And then inhibition of NKHD1 in mice in the descending aorta. Descending aorta are known to be exposing to high shear stress. Downregulated enos and prostacyclin synthase significantly. So this comes in agreement with in vitro work. Now the question is whether NKHD1 controls enos and prostacyclin synthase at transcription levels or at mRNA stability levels. So I performed time course experiment, and in this experiment I stimulated cells with shear stress at different time points, and I assessed the target genes, KLF4 and KLF2 were transcription factors that upregulate enos and prostacyclin synthase. This is well known in the literature. Enos came after, and by the 72 hours, all genes were significantly upregulated. Because NKHD1 came after enos, this means that enos perhaps not primarily dependent on NKHD1 in terms of the transcription, but perhaps it controls enos mRNA stability. So I performed the RNA stability assay, and inhibition of NKHD1 reduced the half-life of enos mRNA. Because NKHD1 is RNA binding protein, I wanted to assess whether NKHD1 has direct interaction with enos and prostacyclin synthase. So I precipitated NKHD1 with specific antibody that can precipitate NKHD1 together with the bound RNA. Then I performed Western blotting to assess whether my pull down worked, and as you can see, with NKHD1 antibody, I could successfully precipitate NKHD1, and from that I could proceed to extract the bound RNAs, enos and prostacyclin synthase. As you can see, enos and prostacyclin synthase were significantly precipitated with NKHD1 antibody. This indicates the direct binding or the direct interaction between NKHD1 and enos, NKHD1 and prostacyclin synthase mRNAs. Looking at functional studies in mice, so I macroscopically inspect the aortas of mice, and as you can see, aortic, we know already that nitric oxide and prostacyclin are important in controlling vascular diameter and vasorelaxation and thrombosis. In NKHD1 knockout mice, the aortic diameter was narrower when compared to the wild type. So I measured the aorta, and it indicates that in NKHD1 knockout mice, the aortic diameter was significantly narrower compared to the wild type. This wasn't because of any differences in the body weight between the mice. And as you can see, thrombosis is seen in the NKHD1 knockout mice. particularly in the low shear stress areas. Areas are supposed to produce thrombosis. Yeah, thrombosis frequently seen in the aortas of ANKET1 knockout mice, whether this is because of reduction in nitric oxide and prostacyclin, or whether there is any other mechanism, it's currently unknown. I looked at also the RNA-seq data, and I found that also tissue factor was one of the genes that has been upregulated when I silenced ANKET1. So this was confirmed in human endothelial cells. So to conclude, I found that ANKET1 was expressed in the vascular endothelium. It was upregulated by vasoprotective high shear stress. It binds directly to enos and prostacyclin synthase and enhances their expression under vasoprotective condition. With that, we could say ANKET1 promote anti-thrombosis, anti-inflammation, vasorelaxation, and protect from atherosclerosis. I have more than this, but it's because it is only 15 minutes. I would like to thank my supervisors in the University of Sheffield, Dr. Maria Fragadaki, Professor Paul Evans, and Professor Sheila Francis, my sponsor on the top, King Abdulaziz University, and the collaborators from the Imperial College London. And I would like also to thank the Immunology Unit for having me to extend my work. Thank you so much for listening.
Thank you very much. Thank you very much, Dr. Rees, for the outstanding lecture. Now, it's my pleasure to introduce our next speaker, Dr. Latir Al-Kirati. She's an assistant professor in hematology and coagulation in the Department of Medicine, Laboratory Science at King Abdulaziz University. Her research focuses on the mechanism of thrombosis and hemostasis. She will be presenting the important topic of the role of fibrin clot structure in the thrombosis. Dr. Latir, we are honored to have you here today. The floor is yours.
First, I would like to thank the organizing committee for inviting. Today, I will talk about the role of fibrin clot structure in thrombosis. First, I thought to give you a brief introduction about hemostasis. So, in case of vascular injury, platelet will be activated. So, in case of vascular injury, platelet will be activated, and that will activate thrombin activation and fibrin formation. So, first, thrombin will cleave fibrinogen to fibrin, and then that will cross-link by factor 13. After that, that will form stable fibrin clot. Then, when stable fibrin clot has done its job, the plasmin will trigger that clot degradation. So it's a balance between clotting and fibrolysis. Overclotting can cause thrombosis. 1 in 4 people die from thrombosis. So what is the thrombosis? It's occur when a thrombus forms inside the blood vessel, disrupting the blood flow. For example, here, arterial thrombosis and venous thrombosis. So what is actually the thrombus? Thrombus contains fibrin fibers, platelet, RBC, and leukocyte. Each component will have a specific percentage depending on each disease. So what's the effect of platelets and clot structure? First, alpha-2 beta-3 fibrin interaction, mediate clot contraction, increase clot stability. Next, the BS exposure and the surface of platelets will trigger the coagulation, which in turn support the thrombin generation. Next, the GP6 fibrin interaction, which increase the Bruchal platelet and promote thrombus growth and stability. After that, RBC. So it's generally thought that RBC is not really involved in thrombosis because all the attention goes to the fibrin and platelet. However, let's see how the RBC can change the clot structure. So during the clot contraction, RBC become polyhydrocyte, as you can see here in the figure. This is the first one, the bioconcave normal shape of RBC, and the polyhedral shape of the RBC using different techniques. And we can clearly see the polyhydrocytes and thrombosis. This is important because it will reduce the thrombus permeability, which in turn will affect the clot lysis. Other mechanisms increase blood viscosity, aggregate, adhere to vessel wall, promote thrombin generation, and increase the thrombus size. Now I would like to show you an example of healthy fibrin clot phenotype on the top and prothrombotic fibrin clot phenotype on the bottom. As you can see, the prothrombotic phenotype characterized by low permeability and low fibrous diameters, also low pore size, which is the area between the fibrin fibers. Also, if you look at the turbidity curve, we can see the prothrombotic characterized by faster clot formation, denser clot, and prolonged lysis compared to the normal formation and lysis. So, prothrombotic fibrin clot is associated with so many diseases like cardiovascular disease, thrombosis, inflammatory disease like rheumatoid arthritis, diabetes, and more. And the thrombus structure is important for risk of thrombosis and thrombus removal. We published this paper last year. We found that fibrinogen level and clot probability identify patients who benefit from catheter-directed thrombolysis after DVT. Also, other groups shows that clot structure can predict thrombosis recurrence and also the post-thrombotic implication. So we can say that clot structure could be a novel tool to predict thrombosis and its implication. So what's new about the clot structure is fibrin film. What is the fibrin film? Fibrin film was discovered in 2018. It's when the fibrin forms sheet-like structure on the surface of the clot, as you can see here in the top scanning microscope image, and the confocal image clearly show the sheet-like structure on the top of the thrombus. And also, we show that fibrin film on the top of thrombi obtained from abdominal aortic aneurysm, and also thrombosis mice model. So this confirmed that fibrin film was present also in vivo. Other groups also show that fibrin film on the surface of thrombi obtained from myocardial infarction and also from patients, ischemic stroke patients. So what we know about the film, fibrin film helps retain blood cells and prevent bacterial infection. Also, fibrin film on ischemic stroke thrombi impairs the fibrolysis. However, nothing is still known about the mechanism the mechanism of fibrin film in thrombosis and hemostasis. So we have a lot of questions. Is the fibrin film prothrombotic or antithrombotic? This leads us to our aim to explore the relationship between fibrin film and inflammation, RBC, platelet, and fibrolysis activity. We published this study 3 months ago, and I would like to explain it now. So, to achieve the first aim, the effect of inflammation on fibrin film, we induced the inflammation in humans treated with 3 different inflammatory mediators: LPS, TNF-alpha, and IL-1 beta. And we showed that all endothelial cells treated with inflammatory mediators show high SCP, which is one of the pro-inflammatory markers. And all of them also decrease the thrombomodulin anticoagulant compared to the control. And only TNF-alpha increased the tissue factors compared to the control. So next, to analyze this further, Hugh cells, as you can see on the top in the green, and the clot in red on the middle, and fibrin film on the surface of the clot. So we use the cell culture confocal microscope to analyze the clot density, and fibrin film was measured by scanning microscope. And you can see here figure B showing the endothelial cells in the bottom and the clot on the top. So the clot density was higher in only TNF-alpha treated cells compared to the control, and also the fibrin film was decreased only in TNF-alpha compared to the control cells. So to further investigate this, clot formation on top of endothelial cells was measured by turbidity assay. So on top we triggered the clot with thrombin and calcium, and in the bottom we just triggered it with the calcium, and we found faster clot formation in inflamed endothelial cells compared to the control. So clots on top of inflamed endothelial cells form faster with higher fiber density and less fibrin film coverage. So we want to investigate the role of tissue factor in relating film development. Cells were treated with small interfering RNA, siRNA, to retard the tissue factor and reduce its expression. And we found, we did the same experiment again, but here we add the siRNA, and we found that pro-inflammatory endothelial cells environment reduced the formation of fibrin film via tissue factor dependent mechanism. Next, we want to see how the effect of RBC in clot coverage. So blood clots were formed in different hematocrit percentage, 10, 30, 50, and 70, and all blood clots within the range of hematocrit used develop fibrin film, as we can clearly see it in the surface of the clot. However, blood clots formed with 10% hematocrit show less fibrin film coverage than those with a higher hematocrit. So we conclude that RBC support fibrin film coverage. Also, fibrin film reduce the platelet interaction. If you can see here in the green box, clot without film and clot with film. So clot in red again, and platelets in green. And you can see the platelet adhesion was decreased in clots without film compared to clots with the platelet adhesion was decreased in clot with film compared to clot without film. And also, we did the experiment using platelet with FT-peptide, which is inhibit the platelet and fibrinogen interaction. So as expected, we found there is no significant difference between clot without film and clot with film when we use the inhibitor. So it seems like the film have the same effect as the thrombin, as the inhibitors. And to also confirm the result, we used recombinant fibrinogen, gamma prime fibrinogen. Just I want to tell you what is gamma prime. Gamma prime is fibrinogen, but without the binding site of the platelets. Okay. So again, we did clot without film and clot with film in wild type, and similar result as the purified fibrinogen. The platelet binding was reduced in clot with film compared to the clot without film. When we use the gamma prime, because there is no interaction between the platelets and fibrin, there is no significant difference in the gamma prime fibrinogen. So the area of the platelet binding site was reduced on clot with film, likely because the platelet fibrin binding site alpha 2 beta 3 is not exposed on the fibrin film. Okay, so this is my last finding about fibrin film and where's the fibrinolysis. So we found clot with film again and clot without film, and we add the TPA. So the TPA will trigger the lysis. However, we found, as you can see in the red, clot with the lysis percentage was higher in fibrin clot without film, as you can see in blue, than in clot with film. Also, plasmin generation was reduced in clot with film with increased lag time, reduced peak plasmin and endogenous plasmin generation, and a longer time to peak. So we can say that fibrin film delays the fibrinolysis. So to summarize, we can say that in small numbers, fibrin film will prevent platelet binding, and that's been reduce the thrombus growth. However, in case of the inflammation, the thrombin generation will be increased and fibrin film will decrease. So it's, the thrombus growth will also increase. In case of thrombosis, we found that with ischemia time, a lot of fibrin film will accumulate in the thrombus, and that will delay the fibrinolysis. So fibrin film formation is reduced under thromboinflammatory condition, supporting clot growth through platelet adhesion, RBC support fibrin film. Fibrin film delay fibrinolysis, which could increase the clot stability. And these area, so the film, we can say the film play both roles: prothrombotic in early stage and antithrombotic in early stage, and prothrombotic maybe in later stage of thrombosis. Thank you for listening. I would like to thank University of Leeds, my supervisor there, and all the co-authors that contributed in this study, and also our collaborator, and of course, King Abdulaziz University for sponsoring the project. Thank you.
Thank you, Dr. Lamir, for this very insightful presentation. Our next speaker will be Dr. Iman Mansouri. Dr. Iman Mansouri graduated from King Abdulaziz University, and he then obtained the American and Canadian boards in internal medicine and hematology from the University of British Columbia. She also finished the fellowship in hemostasis and thrombosis and a master's in health sciences. Then she served as a lecturer at the University of Toronto and a clinical hematologist at St. Michael's Hospital. Currently, she is a consultant hematologist at King Abdulaziz University Hospital.
Thank you, Kamal, for this nice introduction. My talk will be about D-dimers. So going back to clinical side, challenges and insight, and I will leave the last part of the talk to talk about D-dimer in a special population, which is sickle cell anemia patients. So Dr. Ghadir has set the stage for my talk by explaining the balance between coagulation and clotting and fibrinolysis, and fibrin being at the heart of this very delicate process. So D-dimer is the breakdown product of the cross-linked fibrin, and as you all know, it increases in thrombotic events, but also in many non-thrombotic events as well. So we know it's very highly sensitive, yet very non-specific. And it was initially introduced for the diagnosis of the DIC, but since then we have now more indications for using D-dimer. So we use it for diagnosing DVT in combination with clinical prediction rules. We use it in scores to try to predict the recurrence of VTE, diagnosing DIC, and then risk stratification of COVID patients, and finally in diagnosing VICT as well. And so in the context of VTE diagnosis and in conjunction with a clinical prediction rule, D-dimer comes as a tool to help kind of reduce the number and cost needed for imaging and anticoagulation. And the idea is that in high-risk patients, a negative D-dimer is really not helpful because you know, example is cancer patient, the prevalence of VT is very high, and so the negative predictive value becomes very low, and the test becomes useless. While in low-risk patient, the negative D-dimer basically rules out VTE, and so by that reducing cost, time, and radiation. Now, in the context of VTE recurrence, there's so many scores out there trying to estimate risk of recurrence, and all of those have in common the D-dimer. Yet, what is important to know is D-dimer is, you know, incorporated in many different ways in those scores. So in the DASH score, the D-dimer has to be measured a month after stopping anticoagulation, but in the HER2 score, it is measured while on anticoagulation. It actually has to be a iodized D-dimer, a point I will go back to in a later slide. So all that sounds great, but unfortunately, it's not. You know, it's more complicated than that, and there's so many challenges with using D-dimer. So, of course, there are so many factors that increase the D-dimer. Age increases it, and there are, you know, adjustment tools or equations to help adjust to the age. It changes with ethnicity, with sex, with pregnancy, with cancer, with smoking, many inflammatory conditions, and with sickle cell disease, and the list goes on and on. Now, as an example, in the context of pregnancy, initially it's thought that D-dimer is basically useless because most of pregnant females will have a high D-dimer. Until we are now using the years criteria, which basically tries to categorize patients in a better way. So instead of doing a CT scan for every pregnant female who comes with a complaint, we're using D-dimer at different cutoffs to try and lower the number of patients who need the CT scan. And so with the years criteria and the D-dimer, CT scans are avoided in about 39% of patients in this trial from 2019. The other issue is D-dimer. It's a very heterogeneous test. So there is 30 plus D-dimer assays in the world. Some are quantitative, some are qualitative. They recognize different epitopes. They are reported in two different units. There is the D-dimer unit and there is the fibrinogen equivalent unit. There's 7 magnitudes of unit, and then some studies or some labs reported with age adjustment while others don't. And so if we put all of these factors together, we have about 28 possible reporting units, making it very hard to communicate between each other. As mentioned, the different assays, so some are agglutination assays, ELISA or lattice agglutination assays. And this table here just show you some of the central laboratory D-dimer assays. So as you can tell, in the methodology, they are different, as we showed. It was in the previous slide. The units are different, the reporting units are different, and then even the manufacturer cutoff, you have 500s and 200s and 0.5 and 243, making it just impossible to comprehend. And finally, even for FDA approval, not all D-dimers are FDA approved for VTE exclusion. So, we never, we worked in so many hospitals across our training, and we never asked what is the D-dimer here, and should I trust it or should I not? And it's, it was really sad when I learned this the first time that you can't trust any D-dimer. There are some that are approved for exclusion, and some are approved for as an aid in diagnosis only. Moreover, D-dimer is not standardized. So any lab test, you have a calibration process before using it, and that kind of standardize the test. This is not the case in D-dimer, so it is not standardized. It is also not harmonized, meaning one level cannot be, one D-dimer cannot be converted to another test. There are currently efforts trying to harmonize D-dimer levels, but it seems that it is more difficult to say than to do, and standardization seems to be a bit even more far than that. Finally, the assays are not interchangeable, and this is very important in clinical practice because one of the very commonly used scores, the HER2-DO score, which we use in risk prediction of VTE recurrence, depends on a VIDAS D-dimer, which is a kind of D-dimer that is available in very few areas in the world. And so if you are using the score and not really paying attention to what D-dimer you're using, you're probably getting a false result from the score. And this graph here just shows you how various the commonly used D-dimer varies with the coefficient of variation here on the side. That you can see how like one line goes far from up to down. Now, this all has a lot of clinical implications, so finding from one study does not apply to another study. You can misinterpret results or over-rely on a result if you don't use a clinical prediction rule, or misclassify a patient. And so I'm gonna spend the last 5 minutes talking about D-dimer in sickle cell patients, since they are a population we see quite a lot. As you all may know, those patients have enhanced thrombin generation, and they have a persistently active fibrinolytic system, and so they have a constantly elevated D-dimer. And certainly this constant elevation makes D-dimer basically useless to use in patients with sickle cell disease. Now, what we were trying to do is explore D-dimer in our patients. So we looked at all patients with sickle cell disease who had a D-dimer done between 2013 to 2022. And then there was 288 unique hospital visits with a D-dimer, at least one. 72% were hemoglobin S, but the rest were other hemoglobin sickle disease. And 20% of the patients already had a VT event, so we're already talking about very high risk population, but only 6% of the patients had another event. The important thing is only 2% of those patients had a normal D-dimer. So you could kind of ask yourself, so what's the point even of sending a D-dimer test in this patient population? If you wasted 200,000 reals on really no information. Looking at D-dimer levels in VTE patients versus no VTE patient. So VTE patient had a mean of 16, while the non-VTE had a mean of 6. Both are very elevated. This was statistically significant as a difference, but if we look at the graph, there is a huge overlap between the levels. So the question is D-dimer does it have a discriminatory power in sickle cell disease? It does, but it is an acceptable power. It's not good and it's not excellent. The area under the curve is 78%, which is fair number, but not an excellent number. Now the question is, as was done in pregnant patients and is currently being done in cancer patients, can we use a higher cutoff, and by that we try and make sense of the D-dimer to be used. And so if we look at different cutoffs here, when you want to look at a cutoff for a test, you look at something called the UDON index, which basically looks at the cutoff where the sensitivity and the specificity are balanced. And according to the UDON index, this is at 5. But as you can see, the sensitivity is only 83%. And this misses 3 VTE events, which is basically not acceptable. And so a better cutoff, or a potential cutoff, is at 3, which has a sensitivity of 1, but a poor specificity, yet you are not missing any VTE events. So we are really just scratching a surface here. There's really a lot to be done to even consider using D-dimer for sickle cell patients in any clinical sense. First of all, a validation of such cutoff needs to be done. And then after that, a clinical prediction rule, in addition to the new cutoff, needs to be tested in prospective studies to have any insight. And so my conclusion is D-dimer should always be used in conjunction with a clinical prediction rule. Without it, it is really useless. It is important to keep back in our mind how heterogeneous and non-standardized, non-harmonized, and non-interchangeable they are. And finally, in patients with sickle cell disease, they always have an elevated D-dimer, and until more efforts are done, we can't really make sense of the D-dimer result at this point. Thank you so much for listening. I would like to ask all the speakers to come for the panel discussion and questions, please. Okay, is there any questions from the audience here? Okay, I have a question. My question is for Dr. Arish. You said that the ANKHD1 control vascular diameter. What about the smooth muscle cells?
Yes. As long as it is a global lookout in the month. So you think about the vascular thickness.
Yeah.
And perhaps it also plays a role in the aortic diameter. as long as it is a global knockout, not excluding the involvement of this smooth muscle cells in changing the aortic diameter. But I think to answer this question is to conditionally inhibit anchage T1, particularly in the endothelial cells, to identify whether anchage T1 control endothelial dependent vasodilation. I hope this answers your question.
Yes, thank you. Okay.
A question from the audience.
I have questions for, as long as Dr. Ali asked by Dr. Hiba, I have a question first to Dr. Ali. Doctor, is it cost effective?
Well, thank you for the question. Thank you for the question, Mr. Majid. Cost effectiveness actually depends on, I do believe nowadays depends when you're actually doing it. If you're doing it back in the days when actually it would cost and would rely heavily on what other medications available on the market, you would find sometimes not to be cost effective. Nowadays, probably when you have actually point of care testing, as I was mentioning, cheaper methods of actually doing the same test with a higher accuracy. I do believe this is a dynamic thing going forward. And whether it's become routine or not, it's a dream for most clinicians and scientists to have the tiny information about all patients. But then we don't look at cost effective alone. We talk about the anxiety you might be causing for these people. I do have an example for that more recently. I wanted to start a patient in Dua because he has recurrent thrombosis and stuff. So I did WES, whole exome sequencing. And none of the things desirable I wanted to be positive or negative I found, but I found different stuff. So now I just look like almost like opening the Pandora box.
Yeah, you go into the ethical obligation. Do you tell them you or not? You get into that ethical dilemma.
You get into that sense, but it's a result that actually you will know it regardless. Sometimes these results are actually being released to the patient directly, not necessarily to the physician. The physician will create a message. But I mean, the genomic part from that perspective, how insurance companies also are being affecting this rule, knowing that I have a SNP, they consider it a mutation. No, please, it's not a mutation. It's just a variation in the population. Sometimes they wouldn't understand that, and they increase the policy a little bit. It would affect the quality of life.
Which comes into another question. Do we routinely test for these SNPs when given to work?
Honestly, I hope not that we actually do. I hope we don't reach that level where we routinely test for these SNPs. It might be case by case, those patients who develop significant bleeding. Right now, if you ask me, should I give a Pixaban or Rivaroxaban, for example, I would just rely on the convenience. This is a once daily dosing, this is a twice daily dosing, based on the clinical trials that say, well, Pixaban has, for example, less bleeding risk in the CNS, the GI. I would rely also if the patient has a gastric ulcer or gastric abscess. But going to the genomics, saying which of which, I showed an example, but still small studies.
Big question here, Victor.
My question is to Dr. Iman. Can we use 1,000 as a cutoff for the dimers for the pregnant lady for like all the trimesters?
In the years criteria, we didn't specify the answer. But it depends on if you have the criteria and then if it's low risk or versus high risk. So I'm not sure if there's any studies looking at trimester specifically.
I have a question.
ايوه دكتور مجد يعطيك العافيه. شكرا للجميع على المحاضرات، الله يعطيكم العافيه. سؤالي دكتوره غدير، why inflammation reduce the fiber and film formation?
Thank you for the question.
So, previous study shows that higher clot formation, faster clot formation will lead to reducing fibril, and we know in the inflammation environment there's a lot of contributors, a lot of thrombin generation and tissue factor that will contribute to accelerate the clot formation. So once it's accelerated, that will reduce the fibril film in surface.
So I have one question, Dr. Adil. Thank you for this enlightening talk. Actually, it opened my areas to different fields of thrombosis as well. But I was wondering if using antifibrinolytic available drug in the market, like amine phosphoric acid or tranexamic acid, actually would affect the biofilm or not, or the fibril film.
It's an interesting question because we know that the anticoagulant will definitely affect the clot structure, but we need more study to study the fibril film. But I assume that because it's the fibril fibers will be affected by anticoagulant, the fibril film will be affected as well.
I mean, those are called antifibrinolytic, so we actually use them in cases of bleeding. So in patients who go through massive bleeding, we do use tranexamic acid, and it has been basically or not yet proved that actually if you have a patient who's developing blood clot, don't use tranexamic acid in the acute phase, use it later, and this might have a relation to the fibril film.
Yeah, this is interesting because we know that accumulation of fibril film will be formed in later stage.
Sorry, I have a question for Dr. Kadir. There are some autoimmune diseases which are associated with a high risk of clot formation. Do you know, for example, if these people like have mutations or these genes or?
Sorry, you mean mutations or in general inflammatory disease?
Yeah, I mean,
So, for example, rheumatoid arthritis, diabetes, all of these have higher inflammatory markers, and inflammatory markers will affect the coagulation cascade, and that will in turn affecting the fibril film as well as the fibril fibers, making the clots very dense and stable.
But there are no genetic studies that show like mutations in fibrinogen or some stuff like that.
There is mutation in fibrinogen, but I'm not aware about the inflammatory disease.
Right.
I'd like to thank all the speakers for their excellent presentation, and thank you for the audience for their questions. And here we conclude this session. Thank you.
Right, Doctor.
Thank you all. We will start the break now. Please visit also the poster, and we will come back around 1. دكتور ماشي خليه يتفضل الغداء. دكتور ابراهيم.
for the first session today. Dr. Imran is currently working as senior scientist and PI at University of Takhasus Jeddah. He's also a adjunct professor at College of Medicine at the Faisal University. He's a beloved person who actually worked with us for more than 5 years. His lab is actually most prominent lab around the world regarding studying RNA modification, especially mRNA acetylation. Dr. Imran, without further ado, the floor is yours. First standing.
Thank you. Good afternoon. Thank you for inviting me to this auspicious and great gathering of minds. Been very stimulating from this morning. I'm here listening great talks across the whole panel of basic to translational research. So I will try to introduce something I believe people are very interested these days is RNA modification. RNA modification is not a very new word, for people who are working in basic physiology. They understand what is RNA modification and what is the importance of RNA modification in general. But for people, for a layman language, I would like to say that people, when they take mRNA-based vaccine, they understand what is exactly mRNA modification. So you might have seen that the classical mRNA based vaccine actually contains the modified mRNA. It does not have uridine, it is pseudouridine. It has been modified at first position of methyl. So this mRNA modified vaccine actually does not induces any self-induction of inflammation, and actually makes a very nice protein that can actually be used for as a vaccine, and that has been proven. Now this has created a buzz in the market of therapeutics that people want to make a lot of RNA-based therapy. What I will try to do is that not go towards the mRNA vaccine. I think it was very well covered yesterday and day before yesterday. I have seen a lot of great posters there from Professor Anwar's lab. But I will try to bring your attention towards the RNA modifying enzymes, why they are important, what is the real role of these enzymes in oncology. That's the ongoing thing what we are doing right now. So my topic is uncovering cancer-associated modifications, focus on RNA acetylation. So I will try to give a glimpse of what is exactly RNA modification, what is the terminology, what is the basic aspect in physiology and oncology of these RNA modifying enzymes, and then try to bring focus towards the RNA acetylation specifically. Now this is my lab. My lab works on epitranscriptomics. We pick up epitranscriptomics like 5 years before. Before that, we were traditional epigenetics guy, but with evolution of the research topic and interest in the transcription, gene transcription, we move on to this particular area. We are now moving into a very different direction with Takhasusi, that we are trying to have develop technologies or small molecules or therapeutic based on ASOs and siRNAs and conjugated siRNAs for different therapies. I have a very nice chemical biology setup also, where we're trying to develop small molecule inhibitors for these new enzymes, which are in as a drug target, and metabolomics. This is something which is very important for us because we are trying to understand the impact of metabolism or metabolic cues on the transcription of a certain gene. So that's what we are working on, the intersection of metabolism and gene transcription. So this is what we do in our lab. So if you go by a classical definition of RNA modification, it is basically a branch of epigenetics, and it is very well known as epitranscriptomics. So these are actually biochemically modified RNA, and let me clear that I'm not talking only about mRNA, I'm talking about all kinds of RNAs. So tRNAs, ribosomal RNAs, long non-coding RNAs, short non-coding RNAs, the messenger RNA, every RNA type is heavily modified. And these modifications have tremendous impact on the outcome of the protein production or different functions. So I will explore a little bit more. So these RNA modifications are very crucial, and if you they regulate a lot of aspects of transcription and translation, which includes splicing, nuclear export of RNAs, transcript stability, and translation of the RNAs. Now, there are more than 170 RNA modifications in different type of RNA species. So you can understand, it's a big area to work on. But there are some favorite ones. Why we are saying favorite one? Because the field is just booming up. The technology is growing for sequencing, antibodies, inhibitors. So that's why we call them as the favorite one. So if you see the favorite one list here, and I will try to come on the other side. Yeah. So I will try to take your attention here. So the most famous one is the M6A, where you have a methylation on adenosine at 6th position of mRNA. And then we have M1A, M5C, M7G, and then A2I. So if you see, most of the time adenosine is being modulated. And then this is a very specific one. This is the only known acetylation modification. So the mRNA, in this case, get acetyl group on the 4th position of the cytosine. Now, all these modifications have tremendous impact on the mRNA to protein conversion. This is a very unique one. This has been, I already told you, been utilized for making the messenger RNA vaccine. And then this is A2I, where adenosine has been converted into inosine, and then it results in either a truncation or I can say a phase shift mutation based on that. So this is very interesting. Now, if you see these are actually writer, readers, and erasers proteins. So as I told you that some of the modification, the whole machinery is known. So metal 3 and metal 14 are enzymes. They come up with this big assembly, and they conjugate the methyl group here. Then there are the readers. Readers, what does they do? These readers actually find out the exact position where this modification is going to happen, and these are the erasers. So once the modification is being utilized, it has to be erased. It's a normal phenomena, and it happens almost all the process of the journey of mRNA formation till it is converting into protein. And also, if the mRNAs are not converting into protein and they are being retained in nucleolus, they also are being heavily modified so that their half-life stays there. So moving on, this is a very simplistic representation of the journey of RNA. If you see here, the messenger RNA has been formed, and then it requires a modification, and so that it can be easily and safely transported out from the nucleus. So what modification is happening here? It's M6A, and then it goes to ribosome. Recent findings have suggested that these modifications are crucial for efficient translation. So if you lose these modification, the mRNAs either slip on ribosomes and they do not fit in. So the translation rate, and there are translation errors that can result in protein aggregations. So these are some new findings coming up regarding the role of mRNA modification. Now, based on the signal, if it is a stress or some other condition, the RNA will be stored or degraded. And this is happening not only on mRNA, also on tRNA fragment. So we have a lot of data on tRNA also, but today I'm just focusing on the mRNA actually. Now, what are the physiological roles? If you see here, the mRNA is heavily modified, and the modification is actually happening on both sides, the 5' UTR and the 3' UTR and the coding region also. And what does they do? Actually, they regulate the splicing, maturation, localization, translation, and degradation, all the aspects of the messenger RNA journey towards a protein. As I said, tRNA is also heavily modified. The tRNA loop is heavily modified. What does it do? If the tRNA modification are important for the structural stability, the maturation, and the codon binding, that is very important. So these three RNA modifications are crucial. Similarly, ribosomal RNA and microRNA, long non-coding RNA, and circular RNA. So all kinds of RNAs actually contain these modifications, and that's why this field is very, very fantastic to go in and see that if you are interested in any kind of RNAs, you can dive in and see that what this modification is doing in that context, in a disease or a normal physiological context. Most of these RNAs modifying enzymes are actually mutated in neurodevelopmental disorders. So that tells that they are important for development prospect also. Now moving on, apart from their role in basic physiology, as I told, that they are being very heavily modulated. There are many reports about their role in diseases, including neurological and metabolic diseases. And since my lab focus on cancer, so I put this picture there. So if you see, a lot of cancers have these modifications on the messenger RNA. A lot of different messenger RNA carries these modifications so that they can efficiently translate and make a lot of oncoproteins or other proteins that are required for the growth and survival of a tumor. Now, why we are interested in epitranscriptome? As I told you, we were never been a traditional epitranscriptomic guy. We were always been epigenetics guy. We switched into this field, and why we switched in, I would like to introduce the topic here. So when I was in KU, I used to work with Dr. Hani Chaudhary. Hani and we have been this fantastic program with the Oxford people on hypoxia. So we are very interested in hypoxia, and people who work on hypoxia, they know that hypoxia, through the help of HIF transcription factor, regulates almost all aspects of transcription by modulating these different RNAs. Now, this is one compelling, thing for us to move in the field, because we're already working on hypoxia and epigenetics. Why don't we move into hypoxia and epitranscriptomics? And then we are also working on a very, very famous model of ECM detachment. You know that for primary cancers to move into circulation and do distant metastasis, they detach themselves from ECM. ECM detachment is a very nice model to work, because it actually modifies not only the metabolism of a cell, of cancer cell to survive, but also modifies a lot of gene, transcription. And that gene transcription actually we showed that is regulated by epigenetics. So this is a model generated by us, and you can clearly see here that detachment impacts the histone and DNA methylation, and that regulates a lot of genes which are important for cell survival during ECM detachment. These are some basic papers from us, where we have shown that a lot of stemness and survival-related genes are actually epigenetically regulated in ECM detachment model. So these two models, where we were already working with, we know that gene transcription is heavily regulated, and we want to know more that whether in these conditions mRNA is also modified or not. So that actually pushed us into the direction, and we started with a very simple experiment that we took 2 cell lines of breast cancer, and we grow them in normoxia and hypoxia, and then isolated the total RNA, and then we do site exclusion chromatography, and then we separated the big RNAs and the small RNAs. And we fed all these RNAs for LC-MS based identification of almost 82 different RNA modifications. For the sake of today's talk, I'm just focusing on RNA, so I will not talk about ribosome and small RNA. So I will take so if you see here, a lot of modifications are consistently modified, but I will try and focus on one of them is I. So irrespective of the genetic background of cell lines, hypoxia induces A to I conversion across all different cell types we have tested. I am showing the data here of 3 cell lines only. We have data for almost 23 cell lines. So we have extensive information, and we are in firm belief that if you give hypoxia, it induces a lot of adenosine to be converted into inosine. Now, adenosine to inosine, you can see here, it's very consistent across different cell types. Now, what is this A to I modification doing? It's actually, when you have an A to I conversion in mRNA, it regulates non-synonymous codon changes, it regulates splicing, it supports the mRNA to be escaped from degradation, it also suppresses the immunity by modifying the double-stranded RNA. So this is the classical information about A to I editing. So in a lot of autoinflammatory diseases, the enzyme which is responsible for converting A to I, ADAR, is a family of enzymes, ADAR1 and they are being mutated. So if these enzymes are mutated, you have a lot of self-double-stranded DNA, and that will induce inflammation, autoinflammation. So these enzymes and this A to I conversion is very famous in the field of autoinflammation. Now, hypoxia promotes A to I. Now we want to know exactly, if you knock down these enzymes, as I told you, the ADAR, they have 2 isoforms, 150 and 110. 150 retains in nucleus and 110 sits in cytoplasm. And if you knock them down, cells die. So they are actually, if you do transcriptomics, you will see that they are inducing a lot of innate immune response or interferon levels. So that shows that if you knock them down, there is a lot of double-stranded RNA production that induces TNF-alpha and other things that can kill the cells. Now, what we did, we tried to find out, because A to I editing for us was a challenge, because very few groups in the world do genome-wide sequencing for A to I. So we know A to I conversion is happening, but there is only 2 labs in the world right now. At that time, when we started 3 years before that, we were doing A to I sequencing. So we go to Stanford School of Medicine, And Professor Jin Bailey supports us. And what we did is that we try to find out that globally across the genome, what is happening when A is converted to I. So what we did, if you see the data here, that a lot of cells tend to show that compared to normoxia, there is high editing going on in a lot of mRNAs. And then what we did, we try to find out that what kind of these RNAs are actually. And you have a lot of genes related to stress and related to other pathways. I just put a list of some of them. If you see here, these are the very famous ones. So one is GSR, which is very famous for making antioxidant defense to the cells. That shows that a lot of mRNAs are actually getting edited and their protein is getting modulated in terms of expression. So this is what we understand in case of hypoxia. But what about if we move and see in ECM detachment model? So if you culture the cells which are traditionally adherent to the surface and you don't want them to adhere on the surface and you culture them in 3D cells, they're trying to tend to make, a lot of people say steroids, but I believe they are more aggregates, not steroids, because you are not giving them some other things to make these steroids. So these are basically aggregates in some way as steroids, we believe. And if you see them, they are having a lot of genes which are related, or mRNA is related to survival going up, and also they have a lot of stemness. So CD44, CD93, all these markers are very high in these detached cells. And we have already known that these detached cells have a different metabolism than the attached one. And they are actually making a lot of one-carbon metabolites, folate cycle, that can do methylation. So we have all these ideas in our mind, and then we explore just only M6A. What happened to M6A? Machine. And what we found, that almost all the cells have induced metal 3. Metal 3 is the catalytic unit of M6A. If it is going up, we have a protein data also. It will increase the total M6A on a lot of mRNAs. And if you give these cells the M6A inhibitor, what happens if you just measure this area of these spheroids, they are actually going down. So it shows that M6A, we are doing an unbiased study here. We are not focusing on certain specific mRNAs. We are just doing unbiasedly. What we did is that we treated the cells with the known inhibitor for M6A, STM, and then we see a lot of cells are not able to, increase the growth size. They were reducing in growth. Now you can ask me that what mRNAs are being M6A modified? Yes, we have a data about it, but today, just for the sake of time, I cannot go on that video. We have a lot of information about it. Now, it also reduces the mRNA of certain stemness-related genes. So, and you can see the morphology here a little bit. These are some cells which are aggregated, and they are very tough to do with imaging flow cytometry because of the low capacity of passing by cells. But anyway, we have some data, and we want to show you and convince you that actually M6A and other modification like A2I goes up in both hypoxia and ECM detachment model. Okay. Now, having said that, we looked into other modifications also. And one of the most important modifications we were interested was this enzyme, NAD10. Now, why we are interested in this one? Because most of the modification I discussed with you is related to methylation. There is only one modification currently existing from lower to higher organism that do acetylation is NAD10. So this is the enzyme that propagates acetylation on the mRNA. And this was consistent in hypoxia and mostly in ECM detachment models. So we thought, let's focus on NAD10. NAD10 is being, essentially very important for development. It is being related with growth retardation disorders. There's a mutation in this gene, and there are growth retardation disorders. So it is very essential for growth. So you cannot completely knock it down in cells. Otherwise, cells will die. Or if you have a knock, you don't have a double, knockout mice. Otherwise, mice will not, they will be gone in 3 days, I believe. E+3, they have been dead. Okay. So that's why we try to find out what is happening in terms of NAD10 in cancer cells. And then the story starts from here. So we took a very simple, I would just like to introduce you what is NAD10. So if you see here, this is the structure of the NAD10. It has 3 domains, actually, where the RNA is binding. This is the helicase domain, which is important for the unbinding of the RNA. So these 2 domains are actually acetylation domain and the helicase domain are the most important one, and they are actually involved in oncogenesis. We have now clear data about it. We express these individual domains and see which are transforming the cells, and we know now which one is actually most important for inducing oncogenesis. Now, moving on, we try to find out with the help of the online data sets. Actually, if you see on the left side, almost all the cancer types have higher induction compared to control of NAC10. If you just go to the TCA data sets. And then if you see, and if you go to cell map, depth map, which is a data set available right now for cancer cells, and if you just put the data there, you see, if you put all the RNA modifying enzymes, NAC10 is sitting on the top. So it is actually involved in almost all cancer types. And if you can see here, if you perturb NAC10, it actually kills almost all kinds of cancer cells. So it looks like it is an essential component of oncogenesis across the different cancer cells. So this data was consistent. So we thought we have to move with NAC10 because the biology was not known. What kind of RNA has been modified by NAC10 in cancer cells? Nobody was having any idea at that time. The only paper when we started was published in Cell that actually showed that if you have mRNAs which are modified by AC4C, the mark that has been put by NAC10, they are efficiently doing translation. So the translation is efficiently happening if you have NAC10, if you have AC4C on the mRNAs. So that was a very compelling paper. And then we thought, let's dive in and see what is happening in oncology. Now we put a very simple hypothesis that we will take cells which are showing high expression of NAC10, we will do a simple knockdown. We will not do knockout. We want to do knockdown because we want to see what is happening transiently. Because if you can do it completely, then maybe there are some other factors that can come and do some kind of backup for it. So we want to do it transiently. So we knock it down and we want to see exactly whether it will reduce the growth of the cancer cells or not. And if yes, what RNAs are going down unbiasedly. So we have to do RNA-seq for it. Now, what we did, this is a dot plot. It looks like a very small thing, but it took 6 months for us to have this data, only this data, because the titration of the RNA for this one took a lot of time. The reported paper say 400 nanogram, but we end up having 1,500 nanogram of RNA. So you can understand it took a lot of time. So this is a dot plot that confirms that this is total mRNA. And if you do knockdown, it's completely lost. So we have almost 70-80% reduction in the total mRNA acetylation. So the model looks nice. Cells are dying. Let's go and do an RNA-seq. So we did an RNA-seq. Now this is where the story starts. Now, expectantly, what we are expecting that if you do knockdown of NAC10, cells will die. And because it is forcing acetylation on mRNA, and if you remove this acetylation, cells will lose their half-life and they will be less stable. So we will be only focusing on mRNA or pathways which are actually going down. So when we put them and do the analysis, there are a lot of pathways that come into picture. Ferroptosis, fatty acid degradation, PPAR. But if you go from top to bottom, a lot of them involve fatty acid metabolism, fatty acid degradation, PPAR signaling, fatty acid biosynthesis. So something that has been consistently going on. So we did this in one cell line, then we repeated it in multiple cell lines, and almost 3 or 4 cell lines we did it. It was consistent. So we thought, let's dive in with for fatty acid metabolism. I'm sorry for the changes in the setting right now. So what we did, we selected some of the very down-regulated genes in the act. When you do knockdown, you see there's a lot of reduction in the genes involved in fatty acid metabolism. What are those genes? These are, some of them are actually making fatty acid, some of them are important for fatty acid elongation, which includes ACSLs 1, 2, 3, and 4, and the rest of them. Now, moving on, what we did next, we look into our very simple hypothesis that if you do knockdown, there is reduction of mRNA. Now we try to do a RIP sequencing and see whether the RIP IP can show that the AC4C modification was sitting on the top of the mRNA. If you can see clearly here, there is an accumulation compared to the IgG, a high accumulation of AC4C, and when you do knockdown, it is going down. So you can see all 6 genes have heavily modified AC4C on their RNA. And if you knock down, these RNA should technically lose their half-life because the mRNA will be quickly degraded. So we measured with the half-life of the mRNAs, and then you can see that this one, this one, this one, this one, and some of them are significantly going down. Now, we have a clear idea that some of the fatty acid metabolic genes, which are important for fatty acid elongation, and later on spreading into triglyceride or TG production and lipid droplet production, they are going down. Now we move ahead, and then we try to do a very simple experiment. So we give cells palmitic acid, which is conjugated by BSA to do lipogens, de novo lipogens. So we treat the cells with palmitic acid. And how does it move on? So it will first use this gene to make long chain fatty acid, and then this has been utilized to make fatty acid coenzyme, which has been fed for degradation or lipid droplet formation. Now, this is the evidence that if you give cells BSA conjugated in the presence of the NAC10, you will see a lot of red dot. This is oil red staining. And if you knock them down, the oil red staining is actually almost gone. And this was a consistent data with many cell lines. So it gives us a hint that if you knock down NAC10, it will reduce the mRNA related to fatty acid elongation and lipid droplet formation. And then what will happen? You will lose these lipid droplets, which are being stained by oil red. Now, what we did, we used another very important CD body pi, CD11 body pi. It's an antibody that binds with the lipid droplets. So we did a flow cytometry for it, and in the data it looks, very consistent what we see here with this left panel. Now, if this is true, we have to go unbiased again. We want to know only some of the lipid-related genes were there available, or there is a big spectrum of genes. So we decided to do RIP-seq. Instead of doing RIP-qPCR, we want to do unbiased RIP sequencing. So we treated the cells with the lipid, and then try to give the cells AC4C, and then we do IP, and then we do sequencing, and we look for the peaks which are enriching for AC4C. And if you see, the enrichment is actually happening a lot. If you don't knock it down, a lot of enrichment was happening in 5' UTR. But when you knock it down, the enrichment has been completely, almost like 2/3 enrichment has been lost. So a lot of mRNAs in having a 5' UTR accumulation of AC4C. And this is the motif which was enriched. Now, this is the gene. So this is the screenshot for the genome browser. And you can clearly see that all these genes have peaks for AC4C in the absence of the siRNA. But when you do knock down, these peaks have been lost. It is consistent in almost all the genes which we are looking for. So that was very consistent to us that actually AC4C was sitting on the mRNA, accumulating on the mRNA, maintaining the half-life of the mRNA. And if you do knock down, these mRNAs are going to get degraded very quickly. What was their traditional half-life? They will be degraded much quickly. So from there, we concluded that NAC10 is crucial for fatty acid metabolism at least in breast cancer cells, because we have a lot of data of breast cancer cells. Now, we try to see the correlation of these ones, where NAC10 with the traditional, genes which we see for fatty acid metabolism, and they are all positively correlated in TCGA data sets. So that confirms that they are very much correlated, not only in our cell lines, but also in the patient data sets. Now, moving on, you can ask me that, is there any inhibitor for NAC10? Apart from, doing siRNA-based work, can you do some chemical-based work? So there is only one known molecule, which is actually not an inhibitor, it's a cryptic chemotype that do interference with the NAC10 enzyme activity. Does not allow to bind with the substrate. So it's not a true inhibitor, but at least it has been reported. We choose it, and we did treat the cells and did metabolic analysis, untargeted metabolomics. And what we found, the top pathway was, most of the pathways was related to fatty acid metabolism. So a lot of negatively enriched metabolites were fatty acid metabolism. So that confirms that NAC10 was there regulating fatty acid metabolism in cells. Now moving on, this is the mechanism that we propose. We believe that NAC10 is putting AC4C on these 4 classical genes, and they are important for fatty acid elongation, and they are making lipid droplets. We have also some data that it is actually regulating the lipid-based signaling in mitochondria, but I'm not going to focus on that one right now. Now, one very important thing that comes while we're doing this work, because fatty acid metabolism is crucial not only for cell survival, but also for cell death. So there's a new form of cell death known as ferroptosis that has been, highlighted a lot more right now. So if you see, ferroptosis is a little bit different from the classical apoptosis. You have feature of apoptosis, but you are inducing cell death from oxidized lipids, which is actually accumulating and inducing a lot of stress. Now, when the cells have ferroptosis, there is one specific gene that allows cells not to die at that time, and that is SLC7A11. So this gene maintains the intracellular cysteine pool in expense of glutamate. Okay, so you flux out glutamate and you flux in cysteine in the cells, cancer cells. And why cysteine is important? Because it provides the buffering capacity by making glutathione, reduced glutathione. So ideally, what is happening? SLC7A11 is bringing cysteine, providing the cells to make reduced glutathione to buffer intracellular loss and extracellular loss in both cases. So in our data set, this was very highly enriched SLC7A11. So what we do after that, if you see this pathway, I already showed you that ferroptosis was there, and if you see the volcano plot, a lot of genes, the classical 4 gene signature of ferroptosis, they are down-regulated when you knock them down. Yeah, quickly. Okay, so now what happened? I will just quickly translate here. If you see this imaging flow cytometry, this is FITC conjugated cysteine. So you have low cysteine when you knock down NAC10. You have high loss because you have reduced glutathione loss happening. You have membrane potential damage, and then you have lipid peroxidation. So all these things comparingly say that when you knock down NAC10, you have high loss because you are losing cysteine, which is making, reduced or maintaining a glutathione pool. Now, if this is true, what will happen? ROS will go and oxidize the phospholipid membrane, and you will see a lot of accumulation of phospholipid. We did lipidomics in these cells, and if you can see here, there's an induction of phosphatidylserine which are oxidized. So this is a fantastic data that says that NAC10 knockout not only reduces the pool of GSS, but actually promotes the induction of oxidized phospholipid that compel and move cells towards apoptosis. Now, okay, again, the same thing. We have seen that all the 4 genes have accumulating AC4C on their mRNA, and actually this is the way NAC10 was regulating their half-life. And this is the mechanism as I showed you. So what is the outcome so far right now? We know that hypoxia and ECM detachment promotes RNA modification, mostly M6A and AC4C in cancer cells. And these modifications are actually essential for steroid survival, stemness, and maintenance of metabolic fitness through fatty acid metabolism, at least per se right now. And NAC10, which is the writer enzyme for AC4C, is a master regulator of fatty acid metabolism in cancer cells. Depletion of NAC10 induces cysteine depletion and GSH reduction that promotes ROS production and phospholipid oxidation that result in ferroptosis. So overall, we have convincing data that NAC10 is going to be one of the most important therapeutic target in the field of epitranscriptomics in case of oncology, at least right now. Now, this is already achieved. This is all I showed you today is publishing from oncogene to clinical translation medicine. So we have published in good place. So alhamdulillah, we are happy that all these works have been very highly cited. Actually, we have a few labs in the world who are working on RNA acetylation. And now where are we heading right now? So we have this new thing going on right now. We are focusing on PGC1 alpha right now. Because in our data set, we found that NAC10 is not only regulating some of the genes related to fatty acid metabolism, but also actually regulating the master regulator of mitochondrial migrants. And I told you that we saw there's a fragmentation of mitochondria, there's a loss of ATP, there's a lot of energy metabolism in the natural knockout cells. So we want to pursue exactly what is the mechanism. Now we know that NAC10 is actually, if you knock it down, here, NAC10, PGC1 alpha mRNA goes down significantly, dramatically actually, almost 70 to 80%, I believe. And if you do the RIP-seq, you can see that PGC1 alpha mRNA have a lot of, accumulation of AC4C mark on their mRNA. So now we have this hypothesis. We believe that NAC10 is actually key for regulation of energy needed for cancer cells to migrate and survive during the distance tissue, relocation for metastasis. And that's what we are pursuing right now. We believe that there is going to be some very fantastic data with our collaborators and from us. Now I would like to acknowledge some very important people. So we are collaborating in Oxford with Professor Adrian Harris and David Moore. And As I told you, we did the sequencing at Stanford. Jill Bay. Most of the data I showed you is from Mahmoud. He is my PhD student. He is now doing a postdoc in Northwestern. And he actually got the postdoc in the same lab who published the first paper in Cell about AC4C. So it was a great honor for us that our students are ending up in great labs, you know. And the other one is Robert Rashid. He's in UCLA now, I think it's UCI. So he's the one who started metabolomics work with us. Thank you. I will try to, you know, I try to cover a lot of things, but there are some fantastic things happening. Thanks a lot. Thanks.
First, I would like to thank Professor Mohammed Imran for his very interesting lecture. And it's our pleasure now to introduce our next speaker, Dr. Farid Ahmed. Dr. Farid is an associate professor of stem cell and leukemia at the Institute of Genomic Medicine Sciences, King Abdulaziz University. With over 20 years of experience in leukemia research, including 8 years in Munich, Germany, he has worked extensively on animal models of acute myeloid leukemia and normal hematopoietic stem cell. Dr. Ahmed's current research focuses on understanding hematopoietic stem cell, drug resistance in leukemia, and the development of novel anti-cancer therapies. He has authored over 40 scientific publications and holds 11 US patents. He is widely recognized for his contribution to leukemia biology and precision oncology. Please, everyone, welcome Dr. Farid.
Thank you, Dr. Hiba, for the nice introduction. And I would like to begin with thanking the organizers for giving me a chance to present some of my work. I'm trying to work on finding what causes resistance in leukemia, trying to understand the mechanisms, and then coming back and trying to find ways how to overcome this resistance. So in the next 10 to 15 minutes, I would like to go through the work and explain what I am doing. So basically, as an introduction, acute myeloid leukemia is a very aggressive and heterogeneous disease of the myeloid cells. It's a cancer of the myeloid cells, and it has generally very poor outcome. So the patients, when they get this disease, they usually have very short life. There has been a lot of development in the recent times. A lot of new drugs have come up. But the improvement in survival has mostly occurred in younger patients, while the older patients still have a lot of improvement to be needed. So there is a question in our mind, and which is, why does AML remain a hard to treat cancer? So I would like to present this slide to you and explain what are the major causes. So the first thing that comes to our mind is the genetic heterogeneity. So it's immense genetic heterogeneity found in AML. There are so many different kinds of mutations. And one of the big scientists in leukemia research in the US once told me that if you subdivide leukemia, acute myeloid leukemia, it would be a connection of like more than 80 different diseases based on different kinds of mutations. So it has immense genetic heterogeneity, which makes it really difficult to understand. And each type of mutation gives a different disease behavior to the patient, meaning that the treatment is affected by the types of mutations there. So we need a lot of efforts to go further and understand the genetic heterogeneity. The other problem which is associated with acute myeloid leukemia is the presence of what we call as cancer stem cells or leukemic stem cells. And these are the cells which are kind of cells which are similar to stem cells, or we call them leukemic stem cells. They replicate, self-replicate and sustain, and they're also resistant to drug treatments, which leads them to have to be persistent even after treatment of the AML patients. These leukemic stem cells sustain somehow, hide themselves in some niche in the bone marrow, and then once the treatment is over, then they come back slowly and then cause relapse of the AML. Associated with the same thing is the presence of or the acquisition of targeted therapy resistance or chemotherapy resistance. So even the new treatments which are like targeted therapies, we are getting a lot of resistance cases in AML. There are cases of immune, there is a probability of immune evasion for the leukemic stem cells or leukemic cells, which is adding to the complexity, another layer of complexity to this disease. And another complication is the old age of the population. So usually, as you know, that the reporting age for this disease is above 65 years, and most of the patients are 65 years plus. So they have other weaknesses which makes it complex for the clinicians to treat them, which we call it as other comorbidities. So this is what we call as hallmarks of AML. So as I told you in the previous slide, that drug resistance is a major factor or a major challenge in the treatment of AML. So my work focuses on drug resistance. And as I said earlier as well, that presence of leukemic stem cells, which are persistent even after treatment, they bring back the leukemia and cause relapse of AML patients. So that makes it very complex for the treatment. So as a summary, we need to better understand the mechanisms of resistance in different kinds of acute myeloid leukemia. So with this, our lab aims to understand the mechanism of resistance using a multi-omics approach. So we use different kinds of techniques to understand the mechanism of resistance, and then how to try to understand how to reverse the resistance. And that will be the focus of my talk. So, and then we try to develop new, or try to find new targets and develop mechanisms to reverse the resistance. So we also use certain currently being used drugs for other types of cancers or other types of diseases and use them as trying to reverse the resistance. So what I'm going to present now is there are two types of resistance models that we are working on. One is the resistance to chemo, which you see on the left side, and the other which we are also trying to model resistance to targeted drugs. And I'll give example of venetoclax, which is a key drug now in AML, and we try to generate resistance and understand how it develops. So the first model where we generated resistant AML cell lines, we generated resistant cell lines to cytarabine, which is one of the key drugs used in AML, etoposide, mitoxantrone, and aclamycin. So due to lack of time, I would like to only explain one of them, which is aclamycin resistance. So we generated aclamycin cells, resistant cells using K562 cell lines. We exposed them to increasing doses of aclamycin and made them resistant over time. And then we selected individual clones. We didn't collect the whole thing which was resistant, but selected individual clones either by doing single cell sorting, by flow, or by making colonies assay where we pick single colonies. And here you can see some of the clones that are isolated. We have given them different names. And then we try to understand, characterize them, what happened inside, and then try to understand the mechanism of resistance. So the aclamycin resistant cells, which you can see here in the graph, they are resistant. For example, the IC15 K562 parental cells was 154 nanomolar of aclamycin, while it increased to more than 7-fold to 1205 nanomolar, or to much more, as you can see in the subsequent rows. So they have higher resistance to aclamycin. They're not killed easily. So we tried to understand this, and we did microarray using conventional microarrays, and then found out that one of the key genes which has come up is ABCB1, which is actually a transporter gene. And what it does is it just pumps out the drugs from the cells and throws it out. So there is less concentration of drugs inside the cells to kill the cells. And not only that, the cells became resistant to multiple different drugs, which are similar drugs or also related drugs, and sometimes even to for example, to etoposide, which is not structurally related to hydromycin. So both the clones you can see here are highly resistant. So this is the fold resistance shown here in the table. So they were like 7-fold or 10-fold highly resistant to hydromycin than the control. So there are multiple mechanisms which the acute myeloid leukemia cells can derive resistance from. The first one, which is drug efflux. There can be also target modification, mutations, and so on. And there can be also alterations in apoptosis pathways and so on. So when we went back to these hydromycin-resistant cells and did CGH array, we found that there is amplification in chromosome 7, and which is the place where ABCB1 is. So we try to find a way where we use flow cytometry to detect the compounds which can block the efflux. And the top panel shows sensitive cells. If you stain them with a dye called R6G, they stain highly positive for it. While when you have the resistant cells, which is in the middle panel, they efflux the dye and you have less stain inside. So that's why they're lower in the red graph, which is in the middle. And then when you have a test compound added to it, the graph shifts to the right. And this is how we screen for multiple compounds and find out some of the compounds, like here CZ415 or NVPCGM097 or SS574. These are the compounds which are novel compounds or compounds which are used somewhere else, and we found them to be highly effective in blocking the efflux. One of them was rapamycin. So we used different doses of rapamycin and each blocked the efflux of the drug, of the R6G. And not only that, the rapamycin use of rapamycin reversed the resistance. You can see clearly in this figure the two resistant cells. When we add rapamycin, the IC50 of the graph goes down, indicating that they were made sensitive again. Not only that, they showed induction of apoptosis when we combine rapamycin and hydromycin together. And also an arrest in the G2M cell cycle when we combine hydromycin and rapamycin. So we tested some patient samples, and they were like some patient samples which were exposed to hydromycin alone or hydromycin with a homologue of rapamycin, which is called everolimus. And what we found is that some patient samples did have an additive effect, but some didn't have. So the ones which had the effect, which is in group A, had high expression of ABCB1. So it correlates with our hypothesis, and that we can, whenever ABCB1 pumps out, everolimus can block or rapamycin can block the efflux and then be beneficial. So it would be very good to combine these in patient samples. So let me speak about the next phase of resistance model, which is venetoclax resistance. So venetoclax worked by going and binding to PCL2 protein, which replaces the BIM, and then they go and form the apoptotic pore. The bags channelize, the bags and the bag homodimerize, and then they let the cytochrome C escape and initiate apoptosis. So in our case, when we had the venetoclax resistant cells developed in our lab, we found that they have lost bags. So there is a deletion of bags, a genomic deletion of bags happened, and then there is no apoptosis. So these cells are highly resistant to apoptosis. So you can see that there is all the clones that we have isolated, single cell clones, they were all highly resistant to venetoclax, and you can see the resistance levels 100 plus, sometimes more than 300. So they were also resistant to other inhibitors, which are like pan-BCL2 pathway inhibitors and MCL1 inhibitor. So what we did next was to do phosphoproteomic profiling and to understand what changes the venetoclax resistant cells have so that we can find new targets. And this is a kinase perturbation plot where you can see on the right side on the top that there are certain proteins which have come up as to be upregulated in all the resistant clones. And these were actually MAP kinases ERK1, ERK2, mTOR, and CDK5. So they were upregulated in all the venetoclax resistant cells. So we wanted to target them because these are the these are the proteins which are highly phosphorylated in the resistant clones. So when we use the inhibitors, you can see that all the inhibitors have same effect, and there is no difference between the resistant and the sensitive cells. So it means that these inhibitors which we selected now work very well with all the clones as well as the sensitive cells. Which means that when we target the MAP kinase using MAP kinase inhibitor, they work very well. So we tried certain other inhibitors like FLEG3 inhibitor, which is called biltetinib, which is very widely used in AML. It shows resistance in the resistant clones, so it is not a good option. But on the other hand, the right panel, which is sorafenib, it equally kills all the cells. So the IC50 is very similar, which means that this is a drug of choice where venetoclax resistance is present. So that's why. So whenever we have resistance due to the intrinsic pathway involvement, we can target the extrinsic pathway. And this is the hypothesis of this work. So we also tried to screen some novel compounds, and one of the compound that is called INK4, which is generated by the Faculty of Pharmacy here in our university. So we found that it is the cells which are resistant to venetoclax are highly sensitive, in fact, more sensitive than the sensitive cells. So the resistance clones are killed very efficiently by this compound. So our future work aims to model this kind of resistance in animal models using xenotransplantation models or PDX models, which is very very nice. So as a summary, I would like to summarize that rapamycin, as you saw in the beginning, reverses ABCP1 mediated multi-drug resistance. The second part of the talk was that loss of BAX expression leads to loss of intrinsic apoptosis. And using multi-omics approach, we found that MAP kinase and CDK5 is highly upregulated in venetoclax resistant cells. So therefore, when we found that, then we targeted them with inhibitors, and we found that they're really effective. And we also found that EOS4, a new drug developed in our university, is targeting venetoclax resistant cells, and we are doing further studies. Also doing CRISPR-mediated target validation for this new compound. So future studies, of course, as I mentioned, will be to study patient-derived xenograft models for drug resistance, and also perhaps using 3D models for drug resistance. With this, I would like to thank the people listed here. I'm very thankful to all of them and the team here to contribute in this work. Thank you so much.
Assalamu alaikum. So again, thank you so much, Dr. Farid, for the interesting talk from a clinician who treats acute leukemia. This was truly amazing talk, and I hope, inshallah, most of what we did actually we're gonna proceed more forward in actually getting this drug, even the one that's built in the pharmacy, college pharmacy, inshallah, in the markets. Allow me to introduce the next speaker for today, Dr. William Sulami. He's an assistant professor of hematology and at the Department of Medical Laboratory Sciences, Faculty of Applied Medical Science, Cambridge University. I'm the hematology lab consultant, active researcher at the HRU, King Fahd Medical Research Center. She has recently joined the Institute of Genomic Medical Sciences as the head of the scientific departments. Dr. Sulami, mashallah, has unique expertise in next-generation NGS technologies, particularly whole genome sequencing, hematological malignancies.
Assalamu alaikum wa rahmatullahi wa barakatuh. Shukran, Dr. Rahman. It's my pleasure to talk today about the NGS-driven insight in hematological malignancies. And today I will be focusing on challenges and opportunities, hoping to inspire the young scientists to fill the existing gap and also to invite experts in the field for future collaboration. So NGS, or next generation sequencing, has become very famous now, and it's widely adopted in both in clinical diagnostic as well as research labs. So the technology was emerged around mid-2000, like in 2006, as the second generation sequencer, following the introduction of the first generation sequencer that was developed, the Sanger sequencer that was developed like late in the 70s, and preceding the third generation sequencer that was developed like in 2010, as well as the nanopore technology that was developed like around the 2015. So NGS has transformed the genomics by allowing So the NGS has transformed genomics by allowing multiplex sequencing, like multiple samples, and increasing the speed while reducing the cost as compared to other conventional techniques. So NGS offers different solutions and techniques. For example, the targeted design panels of known list of genes and regions, as well as the whole exome, which capture like around 1 to 2% of the genome, like covering only the coding regions and the exon, and also the whole genome sequencing, which covers the entire genome, or at least 99.99, and also offers the RNA-seq or RNA sequencing, which allow like whole transcriptome analysis, as described by the by the great talk of Prof. Muhammad Khan, Imran Khan. This table actually compares between these techniques, highlighting the differences in coverage, average depth, and the type of mutation detected by each technique, as well as the data output and turnaround time and cost. So when selecting between these techniques, I think the right selection highly influenced by the specific goal of the research experiment or the clinical application. For example, the targeted panel is widely adopted in clinical diagnostic because of the cost effectiveness and the high turnaround time, as well as high average depth coverage, but it covers limited number of genes and regions, while as the whole genome sequencing covers the entire genome, identify all types of mutations, but at higher cost and longer, or like, let's say slower turnaround time. In Saudi Arabia, hematological malignancy rank among the most common type of cancer. For example, like non-Hodgkin lymphoma is the 4th most common cancer in Saudi Arabia, and leukemia is the 5th most common cancer in Saudi Arabia when counting both male and female. However, in male adult, non-Hodgkin lymphoma is ranked among the 2nd most common cancer in adult male Saudi, while the leukemia is ranked like among the 1st common type of cancer in children in Saudi Arabia. So hematological malignancies are a group of neoplasm characterized by clonal proliferation of hematopoietic cells at different maturation states, accumulating or infiltrating, for example, the blood represented by leukemia, or the lymph node represented by lymphomas, like non-Hodgkin lymphoma, or the bone marrow represented by the multiple myeloma. All hematologic malignancies stem from genomic alteration, which have been implicated in the clonal evolution of the disease. So understanding the clonal evolution of hematological malignancies is very important to understand the disease pathogenesis. While the exact etiology is unknown, hematological malignancy may arise through the sequential accumulation of genetic events following cell damage within the somatic cells, transforming normal somatic cells into malignant cells. And it's essential to differentiate between the driver mutation and the secondary passenger mutation to understand the disease pathogenesis. A second scenario has become recently very accepted, which say that the first, which proposed that the first head that may predispose to cancer is inherited in the germline, in the germline cell as the pathogenic single nucleotide polymorphism, inherited SNP. And the molecular pathogenesis of hematologic malignancy, as well as the heterogeneity of hematological malignancy, as discussed by Dr. Ahmed Farid, necessitate complex, comprehensive analysis of the genome. So NGS has helped refine classifications of hematological malignancy, for example, the classification of myeloid and lymphoid neoplasm, updated in 2022. And NGS has also help to guide the clinical management of hematologic malignancy toward better diagnosis, prognosis, and treatment options. For example, diagnostic biomarkers include gene fusion, like BCR-ABL in chronic myeloid leukemia, as well as ETV6R in acute lymphoblastic leukemia. Also include rearrangement, like MYD88, or single nucleotide variants like 83, both detected or identified in lymphomas. Prognostic marker like TP53, either as monoallelic or biallelic, can help or to stratify risk assessment or to predict the poor prognosis in the patient. Also can reveal clonal evolution or enable sensitive minimal residual disease monitoring. And therapeutic biomarker guide the targeted therapy, for example, like FLT3, and also assess resistant mutation. The figure shows all three types of genomic alteration identified in hematologic malignancies, for example, like the gene fusion, include the transcript changes fusion, for example, like including the used to be balanced translocation, which is very common in hematologic malignancy, as well as rearrangement, which includes a large structure variant, including duplication, inversion, deletion, large copy number alteration, and mutation, which include like small mutation like single variant or insertion deletion, as well as frameshift deletion. So as we mentioned earlier, like while the targeted panel is widely adopted in clinical diagnostic, but whole genome sequencing as single approach can actually identify all type of mutation in a single experiment. And it's important to emphasize the role of genome study and earlier genome-wide association study that were performed using the earlier microarray technology as the foundation or the groundwork for NGS-based clinical testing. For example, whole genome, let's say, or genome-wide studies can identify novel biomarkers. These novel biomarkers can include coding or regulatory elements within the non-coding region, either in the tumor cells or in the germline as a pathogenic SNP, and can validate the clinical relevance of the variant, for example, by differentiating between the driver and passenger mutation to understand the disease pathogenesis. Also can characterize mutational signature to pinpoint cancer-specific mutational patterns, for example, to differentiate between mutational signature specific to cancer type or, for example, other mutational signature like the aging, and also can be used to can apply comprehensive annotation and allow integration of multi-omics data. So these type of studies are largely supported by large national genomics initiative worldwide, and I think the most prominent studies are these that performed by the United Kingdom, Genomic of England, United States, and the Danish. Inshallah, come in the future, Saudi Genome Project. However, the impact of these studies, the prominent studies, is often limited by the lack of ethnic diversity, which also can limit their adaptation for precision medicine, especially that hematological malignancy are clinically and biologically heterogeneous, and increasing evidence support the environmental and hereditary factors as they may play important role in disease pathogenesis. So, following the success of my field, my supervisor, Prof. Anna Shu, and Dr. Pauline Robb led the largest comprehensive whole genome analysis of 485 CLL patients, and under the Genomic of England partnership. And the study provides one of the most detailed genomic analysis by applying different bioinformatics tools. For example, telomere analysis, mutational signature, and identification of copy number alteration. Like a few years ago, these bioinformatic pipelines were not available. However, now they all publicly available, which can help to validate these results in another cohort. For example, like Saudi cohort in the future. So the study revealed novel clinical and biological insight in CLL, supporting the efficacy of whole genome sequencing in genomic medicine. And while there has been limited number of studies like NGS publication on hematologic malignancy as compared to inherited disease NGS-based study in Saudi Arabia, recent study revealed distinct molecular profiles as compared to global data. And we have also, for example, 3 of these studies actually were conducted by Dr. Hibal Khattabi. And for example, the evaluation of TP53 expression and mutation revealed high prevalence of TP53 compared to many global datasets. And recently there was one of the first whole genome study presented in ASH 2024 by Manar al-Faham. And they actually showed the whole genome sequencing also revealed different profile than when compared to the Western population. We also have additional ongoing study. If you recognize this figure, it was taken from Ragad and Afnan Boster that was presented today, highlighting the diversity mutational group patterns with different prognostic group in lymphoma. Also, we are working on the optimization of cell-free DNA as an alternative as a non-invasive alternative for the FFPE for the clinical management of lymphoma patients. So there have been major national efforts to support the genomic medicine. For example, the recent CAST initiative to call for the centralized biobank on genetic material, as well as the biotech strategy. However, there are few technical, scientific and technical challenges that need to be addressed to explore the potential of Saudi genomics. For example, on the scientific side, I think one of the challenges is to address the gap between genomic versus genetic studies, for example, whole genome versus whole exome, and to define or to establish reference genome on Saudi population to understand the effect of linkage disequilibrium in parental disomy and consequently effect on the Saudi population. I think we need more than one genome to unlock the Saudi genome. And next, I think one of the challenges is to accelerate the translation of the research, for example, by performing functional analysis and by developing AI-driven models that are trained on Saudi genomic data. And there will be an interesting talk on that, will be presented in the next session by Dr. Khalda. On the technical challenges, for example, I think that one of the main challenges will be to the standardization, either for to standardize the pre-sequencing or the sequencing protocol or the bioinformatic pipeline as well, and also to reduce the reliance on external lab or international lab by improving the logistics in the future. Also, a strong governance with clear regulation and funding mechanisms supporting Saudi expertise in genomic medicine is important to move forward in the future. Thank you very much.
Shukran, Dr. Reem, for the very informative talk about the work about whole genome sequencing and NGS in Saudi Arabia, and also at the hematology research unit here and at the KFMRC. Allow me please to ask the next speaker, Professor Lala Hassan. She's a board certified medical oncologist with ABIM and CTCH certification in internal medicine and medical oncology. She holds a clinician investigator certification, master's in experimental medicine with a focus on molecular oncology. She completed advanced training in breast and GI oncology at McGill University. Professor Hassan is also recognized for her advocacy in cancer survivorship, women in oncology, and physician well-being.
Thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh. It's my pleasure today to be with you. Thank you, Hiba. And Prof. Adil is not here, but I hope my thanks will reach him wherever he is. Thank you, Dr. Osman. Of course, you're a dear colleague. We're working together in King Abdulaziz University Hospital. Actually, you see this third designation, clinician investigator.
After I completed my clinical fellowship, I decided to do a basic degree. Okay, I went to the lab, so I understand completely what you are going through. I spent 2 years in the lab doing flow cytometry, Western blot, immunohistochemistry, and I truly applaud you for your patience because you have to wait till the end of experiment to detect to learn is it valid or not. Okay, that's why I like what is being going here. Thank you very much. You ignited a part of me that I thought went to sleep. Okay. And I hope this is going to be a start for future, inshallah, collaboration. I already know many of you and have worked with many of you, but perhaps we can also bridge the gap. I want to see next year more clinicians here. Okay? Many more clinicians, not only basic scientists, because you add to us and you add to you, though I consider myself one of you, yani, okay? All right. Thank you very much. Go back. So I want to show you how it's important what you are doing in the labs because it is translating into clinical impact. And you're more than welcome to join us in our clinics to see how valuable this basic research, followed by transitional research, followed by clinical research to improve patients' outcome. So we are going to speak about something very important, targeting the target. We're talking about different targets as you see here. First of all, starting with HER2. HER2 story is very, let's say, inspirational. It was discovered in the 90s as a proliferative marker that increases the metastasis proliferation of certain type of breast cancer and make it resistant to chemotherapy. In 2001, the first monoclonal antibody targeting HER2 came into use, and there was a revolutionary improvement in the clinical outcomes of the patient with breast cancer. Truly, if you are a practicing oncologist and you use, we have the first one was trastuzumab, followed by pertuzumab, and not only that, but the story continues to combine this monoclonal antibody with chemotherapy to improve delivery, improve the killing of the cells, and also enhance the antibody-dependent cytotoxicity. Now we have something called antibody-drug conjugates, such as TDM. هذه الشاشة لا بعيد عني. Okay. Now it's TDM and TDX, and we're gonna discuss some of them. I'm gonna go through over the clinical trials, and this is a phase 2 clinical trial, and I'm happy that my colleague Osman is leading the clinical trial unit here. Okay, I don't know why it's not slipping quickly. All right. So basically, right now, we are not only treating the histology or the site of malignancy, but we are looking for certain molecular alteration that will guide us in using certain treatment. That's why we say histology or site agnostic. You know what is agnostic means? ملحد بالعربي, but it doesn't mean not. It means you're treating the molecular alteration regardless of the histopathology. All right? So I don't care if I have patient with cervical cancer, endometrial cancer, ovarian cancer, biliary tract cancer, bladder cancer, or other tumor. Only I care about the expression of HER2. If they're overexpressing HER2, which is usually tested by immunohistochemistry, and I give them the treatment that is called TDXD, which is antibody drug conjugate, trastuzumab, combined to a chemotherapy that is called deruxtecan, then you see how improved is the outcome. The first one on this slide is progression-free survival. The orange line significantly improved in comparison to the ones who are not overexpressing the biomarker, which is HER2. Similarly for overall survival. So where do we use it now? It has multiple indications. We use it for HER2 expressing breast cancer. We have another entity that emerging that is called HER2 low, which is tumor expressing HER2 but not in a high quantity.
it's in intermediate or lower quantity. Also in a gastric cancer and gastroesophageal adenocarcinoma, and in non-small cell lung cancer. Moving to another target, which is BRAF V600E mutation, or actually 600E. So BRAF is responsible for proliferation, cell growth, survival, and usually it's activated when there is a growth factor binding to the receptor, causing ligand-dependent activation of the pathway, reaching to the nucleus and increasing the transcriptor factor responsible for growth proliferation. Sometimes when there is BRAF mutation, specifically V600A, there is constitutive activation, so regardless of the growth factor binding. It's usually seen commonly in thyroid malignancy, up to 50%, also in melanoma and in hematological malignancy, highly prevalent, especially with hairy cell leukemia. How does it change the practice? As you see here, if you have or identify BRAF mutation, specifically V600A, then you get an extremely good response rate, especially in hairy cell leukemia and thyroid malignancy and melanoma when you use BRAF inhibitor. We have multiple BRAF inhibitors. The ones I mentioned here are Dipiravir and Tirametinib is basically a MEK inhibitor. What you're doing, you are also blocking the down cascade signaling that may cause resistance to the BRAF inhibition. So it got approval for histology agnostic in unresectable or metastatic BRAF. So you see, completely different than what we used to practice 15 years ago when I did my fellowship. It's basically, we used to say breast cancer, but now, for example, melanoma or leukemia, but now we say BRAF V600A mutated solid tumors, all tumors that has this mutation. Another biomarker that can be targeted is RET fusions. So we see RET fusion mainly in thyroid malignancy, whether papillary thyroid or medullary thyroid, smaller percentage in non-small cell lung cancer. And we have two alterations, either, I don't know why it's not flipping easy. Sorry for this. This is a clinical trial, again. This trial is called basket trial because you identify a biomarker and you screen patient with different malignancy, regardless of the site of their malignancy, regardless of the histology. If you identify the biomarker, then you enroll the patient in the trial. That's why it's called basket. So, multiple malignancy, as you see here, pancreatic, colon, cholangiocarcinoma, small intestine cancer, sarcoma, and non-primary, ovarian, and even breast. And those patients were treated with RET fusion inhibitor that is called silpicatinib. And you see how beautiful this waterfall plot? Amazing. Almost all responded, except a small percent. Another RET inhibitor that is called the bralecitinib, which is again used in the patients who express this RET fusion or have RET fusion. So again, these medications, silpicatinib, received histology agnostic approval by FDA for metastatic solid tumor with RET infusion and progressive disease on previous line of treatment. Yep, you're right. You saved me. Another target that received the approval for agnostic approval is the NTRK. NTRK infusion or genes, fusion genes, basically seen or expressed normally in non-neural cells or sometimes in cancerous cells. As you see here, it down cascade the RAS pathway and sometimes also PIK3 kinases to enhance proliferation, differentiation, and survival. And usually it is activated in malignancy. We have two types of molecular alteration, either fusion or point mutation. But the target that identified and approved for histology agnostic use of targeted therapy is fusions, because it is the activating and the one that is predicting the response to the inhibitors. Where do we see it? We see it in multiple malignancy, but the highest percentage is seen in salivary cancer and also in a very rare subtype of breast cancer that is called secretory breast cancer. And in other malignancies, such as pediatric malignancies, specifically sarcoma. We have already medication that we've been using that is called intracatheter, used for intrac fusion positive cancer, as you see here, another basket trial, multiple malignancy with one biomarker alteration and one medication for all, and you see how impressive is this waterfall plot. Another medication called larotrectinib. I'm sorry about the names. I don't know how they come up with it. Okay. Actually, there is a basis for a nomenclature. We can discuss it later if we have time. And just to tell you how this is truly significant for us as an oncologist and for the patient. This is a patient who had breast cancer that was tested positive for intrac fusion. You see, previously, the one with very bad, this is CT scan of the chest, showing the lungs. So basically, you see how the lungs is completely impaired, the patient requiring oxygen therapy, and only after 1 month on intracatheter, which is intrac fusion inhibitor, almost complete resolution and no requirement for oxygen therapy. This is life-changing. If you're practicing in clinical oncology, it is. It's going to impact you deeply. Another case, and this one is undifferentiated sarcoma. And maybe you hear that sarcoma is a bad disease. It is a bad disease, unfortunately. Chemotherapy doesn't work. Or it works for a very short period of time, and then it's going to relapse and come back. So this is another patient with sarcoma with spread to lungs. You see how badly the lungs are affected. After cycle 5 of larotrectinib, almost complete resolution of all the lesions in the lung. So this is the indication for intrac that we've just discussed. The pathology, the pathway to histology agnostic approval usually start with identifying a target, but not in all scenarios. Sometimes we have a medication that is working exceptionally in some patients. So we go back to the tissue or blood sample of those patients and we try to identify why this medication worked this way. And then we come up with the biomarker. Alternatively, the usual pathway, which is identify the marker, test the drug, and then if it has a clear benefit, it gets this approval. As my colleagues just mentioned, there are multiple ways to obtain samples, and I want to tell you something that we care about when we prescribe this medication and also when we test the patient. There is always, for each clinical trial, something called companion approved molecular testing. Okay. Some of these biomarkers tested by immunohistochemistry. Some of them tested by NGS. Some of them tested by circulating tumor cell or free DNA cell. So according to the approved companion test that was used in the trial, we usually do this. Alternatively, when we are short of resources and expertise, we do NGS, which is good in identifying all the changes, but it has to be read well. Okay. This table shows the list of approved histology agnostic therapies, and the one that I haven't mentioned is on the top, actually the two top lines, which is the immunotherapy. Pembrolizumab also received histology agnostic approval, but the biomarker we're looking for is what we call microcirculating stable. It's basically we're testing the mismatch repair enzyme, and if it's deficient, then this patient is eligible for pembrolizumab. Same for the second one, which is another immunotherapy. Again, we have to test for mismatch repair enzyme. In case it is deficient, you can use the treatment. The list is expanding. All these medications are being tested right now as we speak for histology agnostic approval. So the landscape of oncology is changing significantly. I remember when I did my fellowship, non-small lung cancer used to have one biomarker change, which is EGFR mutation. But now to treat lung cancer, you have to do upfront NGS because you identify multiple biomarkers. And then if you don't have targetable mutation or targetable alteration, it's not necessarily mutation, but there is fusion, so we can say alteration. Then you go for different type of treatment, which is chemotherapy. So definitely, we're moving slightly away from chemotherapy, but chemotherapy is still very important pillar in oncology. All right. Thank you very much, and I'm happy to be with you.
Thank you so much. I would like to invite the speakers for the panel for discussion. Dr. Farhan and Dr. Khalid. We had a very great talks moving towards different specialties in oncology, either figuring out why the patient does relapse and what we can do in terms of actionable therapies and what we should be doing after the NGS and Saudi, and moving back before actually starting from the lab as well, by developing new drugs and actionable translational research, which we really want in BLAC. I would like to get a chance if any questions from the audience. Have one rising hand.
My question is to the last speaker who tells so many things about effect of chemotherapy and drugs related to the mutational treatments. So regarding the treatment, if you give some monoclonal antibodies, and it has been seen so many times, there are so many side effects of this treatment. And what do you think about the side effect? Why there are so much side effect? So how you can overcome the side effect? Is there any research going on to focus on the treatment of side effect of using all of this kind of antibodies and medicines?
Yes, when we have the clinical trial, also the side effects are reported and how to deal with the side effect. There is guidelines to help clinicians like me manage the side effect. Actually, my role is not only prescribing the medication, but making the journey of taking this medicine easier for the patient. So we have definitely proactive measures to prevent certain side effects. Sometimes we monitor patients closely for side effects to promptly act when they occur. And sometimes we have to deal with it as it occurs. Not everybody will get side effect. It's not universal. You have percentage for every single side effect.
Take the floor for another question.
Yeah, to the same speaker. So I think in your last slides, you mentioned something which I didn't quite catch clearly. I think some fusion event. So is it fusion, gene fusion, or cell fusion, please? Can you explain more, please?
You mean rate fusion or intrac fusion?
Yeah.
Okay, intrac fusion.
Yeah.
It is a fusion that is resulted from translocation between different chromosomes, resulting in a new activating oncogene that result in malignancy. So it is a driver oncogene.
Thanks. Thank you very much for the comprehensive and informative talks. I have 2 points, one for Dr. Amran and the other one for Dr. Farid. Speak of NAT10 as a transcriptive of nuclear factor. I'm curious about the situation of nerve 2 or nerve 1, especially in the depleted cell, the NAT10 depleted cell. Have you looked at that transcriptive nuclear factor?
That's a great question. Not yet, but what we know is that transcription is not a very simplistic layer. It is multiple things have been added there. So I cannot say that this is the only driver. Yes, what we are seeing right now is I just had the new discovery what we have found that if you knock down or remove one of the modification, you will see the landscape of other modification are also getting changed. So that's the new story. We are working on it. And that's not only our lab. We just presented the data somewhere outside, and they also confirmed that depletion impacts the epitranscriptome as well as epigenome. And also, we saw some modifications have been changed at histone. So yes, there are a lot of changes happening, but we cannot consider right now that it's only one way in one direction.
Because I believe that NAV1 kind of regulates the NAV10. So I don't know, I mean, in your lab, if you would go, but based on you're saying, especially the signal cells, that too many and the kind of was talk between them.
So if you go deep dive in the histone acetylation, where there are super enhancers which are controlling a long list of genes, oncogenes. So we do found that in certain cases like glioma and leukemia, liver cancer, so there are some super enhancer signals on the top of the active gene. So yes, I mean there are some upstream regulators for it. We are trying to find out that its correlation with certain super enhancers so that we can try to understand who activate the actin transcription in these carcinomas.
Okay, thank you very much, Dr. Hawan. The other point for Dr. Farid. So you mentioned that when the cells exposed to the venticlax, it develop resistance, and then you're targeting after later on the I believe that the kinase or the phosphorylation. Have you tried to like put the drugs together and see how the behavior of the cell against both of them together instead of individual exposure?
That's a very good question, actually. It's a very good question. We tried also, but there is no point to add venetoclax for combination because venetoclax doesn't work in these instances at all. But we did try, but we found no effect of venetoclax. It was showing resistance.
I have a question to Dr. Lee. Dr. you have mentioned that we can use whole exome sequencing, whole genome sequencing. Don't you think that whole genome sequencing is still early to be utilized in the clinic and it is for research use and mainly in the oncology? Where we are in terms of the clinical application of whole genome sequencing?
Okay, in Saudi Arabia, I think whole genome sequencing is still like could be a potential like research tool because like as a single approach, like it provides comprehensive analysis of the genome, identifying many type of the mutations. And so the cost, for example, like reduced, then perhaps the targeted panel is more applicable for the diagnostic service. However, whole exome sequencing is more successful, for example, like for inherited disease because of the power to identify, or of its sensitivity to identify like single variants. But it's limited when it's come to oncology, for example, as mentioned that there's like lots of large structural variants in oncology, gene fusion, copy number alteration that require, like, for example, either RNA and DNA panels or whole genome sequencing. So the whole exome sequencing is limited.
Thank you, Dr. Green. Yes, correct. It is expensive, but it is available, and it's been covered by insurance. And if you are practicing in a center that is, let's say, wealthy center, okay, they're gonna send it for you. Usually, it's outsourced. What we are doing right now, we're doing NGS with a or exome sequencing. If you're paying out of your own pocket, it's gonna be around 30,000 something.
So do you do it like here in Saudi, or you send it
out? It's sent out. It's sent out. It's all sent out, but it is as an essential part. Why I'm bringing this up? Because we need to have this in-house. Yes, we need to really do it in-house because unless your patient is covered with insurance, so or you're practicing in somewhere that is wealthy, you can do it because truly because. For example, in metastatic breast cancer, I would like to have an upfront NGS telling me what I'm dealing with. Because our treatment right now has changed significantly. Okay, not only the first line, but also later lines, what to give as the first line treatment, or what to keep for later, and it's not always available. So definitely, if we can نوطنها, yes, you can. Great, thank you.
We can, as for example, we have the expertise for it, but we need the infrastructure for it, I think.
And collaboration between clinician and scientists. I think we are reaching the end of our session, and I would like to thank all of our speakers. It was really a wonderful talks by every one of you, and we wish that we have more time to learn and to get from your experience. And the take-home message from this session, as said by Dr. Atlan, we need always to communicate together, and the communication should be between the clinician and the scientist, because from there we can do something really usable in the clinical practice. Thank you all. We have a 10 minutes break. Please come back again. We need your support for the last session of today. There is a very nice talks, and we will announce the poster winner also by the end of that session. يلا دحين نقعد انا ما اجلس Okay, welcome back everyone to the last session of the day. It's my honor to present the moderator of this session, Dr. Imran Khan. He has been already introduced by my colleague, but shortly I will say he is currently working as a senior scientist and principal investigator at the Research Center in King Faisal Specialist Hospital and Research Center. The second moderator, Dr. Hadira Sadoun, she is an associate professor of hematology in the Department of Medical Laboratory Sciences. She earned her PhD in stem cell biology from the University of Manchester in 2016. Dr. Sadoun previously held a position as the Dean of Al-Ghad College, and currently she is a part of the research team at the Center of Innovation in Personalized Medicine at King Fahd Medical Research Center, where her research focused on using vitamin D and ATRA nanoemulsion to enhance the differentiation therapy in acute myeloid leukemia. Welcome everyone.
Okay, good afternoon everyone, and welcome to this session. We're going to have 4 outstanding presentations that will highlight cutting-edge approach in cancer research, spanning extracellular physical, biotech innovation, nanotechnology, and AI-guided precision medicine. Please welcome with us, our first talk is going to be by Prof. Faisal Al-Zahrani. Prof. Faisal is a faculty member in Biochemistry Department at King Abdulaziz University. He's a principal investigator at Stem Cell Research Unit. He earned his PhD from Northwest Cancer Research Institute at Bangor University in the UK. His research interests include stem cells, biology, particularly exosome and their application in disease management, with focus on cancer, infectious disease, and overall well-being. Please welcome Professor.
شكرا جزيلا. بسم الله الرحمن الرحيم، الحمد لله رب العالمين، والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. Thank you very much for the kind invitations and for the introductions from our great moderators. Also, thank you for being patient and until the last sessions of the. Today, I'm convince you about the importance of the exosomes. I will try to convince you about how we should find alternative resources for these exosomes. It will be brief introductions about the EVs, importance of the cellular communication and disease pathology, overview of the stem cells, derived exosomes, and their effect of the exosomes from the MSCs on the HCC progressions. Finally, I will talk about the plant-derived exosomes as a promising source for these EVs. My journey started very simply from very simple organisms, S. bombi. Then I moved to the cancer stem cells research. Then I started my journey on the iPS as a normal stem cells. After my PhD, I found myself in the EVs world. Here in these photos, you will see transmission electronic microscope images of exosome. That the last exosome I isolated with my hand in 2021. And before I started my talk, I should give definitions for the EVs. It is the particles, their size is 32,000 nanometers, released naturally from the cells, enclosed by lipid bilayers and incapable of replications. They act as a complex signaling vehicles that can effectively target the recipient cells through the specific surface molecules. It is now recognized that all living cells have the ability to release these EVs. To be not confused, exosomes historically the most commonly recognized type of the EVs, and the first described in the early 1980s, and it was described as a disposable waste system. And when we talk about the exosomes, usually we have three categories, apoptotic bodies and microphysicals. However, the field is expanded. So we have now oncosomes, autophagosomes, many other EVs. So you shouldn't be confused. And recently, in the last month, polypipisomes in the 2025. So don't confuse if I am using exosomes or EVs, because the field are expanding quickly. If we are talking about cell-to-cell communications, it involves a variety of mechanisms. We are talking about hormonal communications, nervous communications, and lastly, the exosomal communications, which is small vesicles carrying proteins and RNA between the cells. If I would like to divide the communications, it could be a bad sources or a good sources. If I'm talking about bad sources, the most common is the cancer cells. If I'm talking about good sources of the exosomes, the stem cells will be the most common one. Why we are interested about exosomes? There are many applications, including diagnostic uses, therapeutics potentials, and also understanding many diseases. And I would like to give example from our study briefly for each applications. If I'm talking about the cancer diagnostic, exosomal microRNA offer a non-invasive, a sensitive approach for detecting colorectal cancers at earlier stages. The exosomal microRNA are stable and can be detected in various body fluid, such as blood and urine. Also, the unique microRNA profile in colorectal cancer patients can improve the diagnosis and prognosis. And we have one graph here showing how is the number of publications in exosomal microRNA are increased. If you notice here in the 2018, the number of publications in exosomal and liquid biopsies is highly increased. And here is a very simple story that led by Dr. Tahani Baksh, which was a PhD student in the King Fahd Centers, and now she's assistant professor at Jeddah University. We collected the serum from the colorectal cancer patients from the KU hospitals, then isolated the exosome, then doing RNA-seq. Then we identified a microRNA that highly associated with the level of the severity of the colon cancers patients. Not only that, these microRNA are involved in the pathway related to the progressions of the cancers. And also in the stem cells, we use different models to use this to treat different disease, kidney disease, liver disease. And here is one approach that we use. We isolated the exosomes from the cancer cells, cancer stem cells, and from the mesenchymal stem cells. Obviously, the mesenchymal stem cells reduce the tumor size, reduce the cancer markers, and the cancer stem cells doing the opposite effect. And this work has been done in collaborations with Prof. Islam Saaduddin and also under the supervision of Prof. Saleh Kraim. Also, in the infectious disease, we have a lovely story in collaborations with KU Hospital and other hospitals, collaboration with Prof. Imran Khan and other colleagues. We isolated the exosomes from the COVID-19 patients. Then we did untargeted metabolite profiling for the extracellular vesicles, and we find a very interesting data, molecules that are associated with the severity level of the COVID-19 patient, specifically those who are admitted in the ICU at KU Hospital. And also one of the interesting things that we found in these data, we found antibiotics molecules in this metabolite, and when we check the clinical data of the patients, we found this antibiotic has been given to these patients. Okay, this is a quick introduction about what we have done on the exosomes applications. Now, what is the problems? If you look at this, one commercial product using exosomes, and by the way, if you see the amount of the exosome has been used in this small bottles, it is 11 billion exosome, which required hard working. If you are in the field of the exosome, you will know that you will need to grow millions of cells to extract this amount of the exosomes. And the mass productions of the exosome, it is one of the biggest problems in the field. This is why we are thinking about alternative resources for the exosomes. And one of the things we found, it is a good source of the exosome, is the milk, and specifically the Arabian milk. And we did a study with one of our PhD students, Dr. Manal Babakar, and we published a few articles on that. And the number of the study on the Arabian camel milk exosomes, can be counted in one hand. Then we think about another alternative resources, which is the plant-derived exosomes, which is PDVs. The plant exosome, it is edible sources, safe, can be found easily. So we choose the sea berries and garlic exosome. And we are one of the first people to isolate the exosomes from the sea berries and garlic. And as you can see, here is the transmission electronic microscope showing the exosomes, also the protein markers, and the number of the exosome we isolated in this experiment is over 50 billion exosomes, to show that how is the plant, it is a very rich source. Then we test these exosomes with one of our master students, Yasser Rida, and it showed that the inhibitions of the exosomes for the cancers, for different cancer cell lines, specifically for those aggressive cancer cell lines, such as the triple-negative breast cancers. And if you look at some of the survival analysis data, we found that some high expression genes like MAB3K1, MED12, NF1, and others, they are involved in the aggressive tumors behaviors and disease progressions. These genes can be targeted by the garlic microRNA exosomes. So by bioinformatic analysis, not only we found the cancer inhibitions, also we found these genes can be targeted by the microRNA that we found in the exosomals of the garlic and the sea berries. And we did other microRNA regulations work through the bioinformatics, and I don't want to go deep on these details. However, as the plant exosome field is one of the recent things that's happening in the field, the International Society of the Extracellular Vesicles has formed new plant task force working group. So if anyone who's working in the plant exosomes are welcome to join this group, and usually they will produce a guideline. It's published in peer review journal, and usually these guidelines gain a lot of citations, like others guideline which has been published previously. And I don't want to talk more about our work. One recent study, which is published this month, to show that how can we use very advanced technology to improve the myocardial infarctions by producing exosome using Wi-Fi to improve the health of the patients who are suffering from the myocardial infarctions. And in the end of my talk, I would like to share that we have a published book in the Springer Natures, and we are currently editing book regarding the plant exosomes, and hopefully published very soon. And also in the stem cells unit, we announced our latest book about the stem cells, and which included some of the exosome research. So to take home message, the field of the extracellular vesicles is rapidly growing and significantly impact the quality of life and the disease management. Challenges remain in the accurately characterizing and identifying markers for each type, Enhanced isolation technique and availability will be crucial for expanding the innovative and translating research into practice. Exploring alternative, natural, and abundant sources will be the future directions for industrial and pharmaceutical applications. And in the end of my talk, I would like to thank many of our collaborators, Faculty of Science, Biology Department, King Faisal Hospitals, Johns Hopkins University, and CACS, and also the funders for their support. And thank you very much for your listening and attending. Assalamu alaikum.
Thank you, Professor, for the lovely presentation. It was very eye-opening and informative. Please welcome our next speaker for today, Dr. Hind Al-Khattabi. Dr. Hind will present innovative biotech approach around 123 triazole compound for targeted breast cancer therapy.
Assalamu alaikum wa barakatuh. Good afternoon, everybody. I'd like to thank Mohammed Swasol. أحتاج مايك لأن أنا من الناس اللي يتحركوا. Good afternoon, everybody. First, I would like to thank the organization committee at KFMRC for their invitation, and thank you to Dr. Hadid for introducing me. Today it's my pleasure to present my work about the innovative biotech approach of array triazole compounds for targeted breast cancer therapy. I will start with the incidence rate of breast cancer, and I will talk briefly about the stages and subtypes of it. Then we will see the current therapies and associated challenges briefly. After that, we will talk about the design of the novel synthetic compound and the biological studies related to it. So let's start with the challenges of breast cancer. Breast cancer is the most, as you know, it's the most common cancer in women all over the world, and it affects 2.3 million women annually. Also, the incidence rate in Saudi Arabia is significantly very high. As we can see here, in 2001, it started with, or in 2001, the breast cancer was 500 cases, and it increases and reached to 4,000 cases in 2020. This is based on the statistic of the Ministry of Health. Here are the stages of breast cancer, starting from stage 0, where the cancer is, or where it is pre-cancer condition, and all the way to stage 5, where the cancer can spread into other organs of the body. Another challenge of the breast cancer is the heterogeneity. So it's not one disease, it's basically diverse subtypes. And when we say type, it's like the reference of the unique features of the cancer cell under the microscope. There are ductal carcinoma in situ, DCIS, which is the early stage or non-invasive stage, and where the cells may evolve and to reach the invasive breast cancer, and invasive, but the vast majority is for invasive breast cancer, and there are two types. Invasive ductal carcinoma, IDC, which is the most common, and it accounts for 70–80%. Another type is the invasive lobular carcinoma, ILC. They also labeled some types with the receptors. So there are ER, PR positive, which respond more to hormonal therapy, and there are also the HER2 positive. There are triple negative, and it's the most threatening and aggressive breast cancer. The current therapy are, as we all know, surgery, chemotherapy, radiation, hormonal therapy. With all, or despite all progress with the strategies of treatment, breast cancer still or remain a challenge in the clinic due to maybe the drug resistance, due to the limitation of the existing drug, and also because of the aggressiveness of some types of breast cancer. So there are always unmet need for more effective, more precise, more personalized medicine. Today I'm excited to present our design of a new synthetic compound, which is based on benzylaryl acetamide adenine triazole, which might open the door for more effective treatment in breast cancer, and especially for the targeted therapy. Let's talk about the story of designing our compound. As you can see here, there is part A and B. This is the pharmacophore of the compound, and this pharmacophore was chosen because of it was really successfully in the clinically approved drug that targets SARC and tubulin. And because our collaborative member, also the team in the pharmacy college in KAU, Dr. Abdel Sattar, they are really very well aware with the activity of this compound. So we discussed how to improve those compounds. They added first the indazole to this pharmacophore. Actually, the indazole is, they produced the KIM-161. The KIM-161 is a fantastic drug. It showed or demonstrated high efficacy in terms of IC50, but it has a major challenge. Although it's also having hydrogen bonding properties, but it has a major challenges because it's high molecular weight and less solubility, and also it hits as a pain, and this is some defects of this compound. So we discussed a lot about these problems, and they come up with the innovative solution to replace the indazole with the triazole. The triazole actually have the same, or keeping the same hydrogen bonding properties of KIM-161, but with lower molecular weight, so it will be less, it will be more soluble, and we believe that with this modification, it will be more effective, and also the most important is that the triazole is non-painless. With this modification, we came up with those 8 compounds, which are triazole-based compounds. As you can see here, the chemical drawn or the structure of the 8 compounds, which we expect for them more solubility and more response for breast cancer. Now let's move to the biological studies of these compounds. We first measured the cytotoxic activity. And as you can see here, the IC50 of all compounds, they are all effective, but there are two compounds which are compound 1 and 6. They're really more potent, and the IC50 for compound 1 was around or nearly 1 micromolar or less. And the compound 6, you can see that it's 1.5 to 1.8 micromolar with the three, we take the average, of course, the experiments, and these two compounds actually exceeding the reference drug that we use. We use the storyboard as a reference drug, and as you can see here in the dose response curve, compound 1 and 6 treated the MCF7 is more effective than the reference 1 storyboard. Then we evaluate the apoptosis using the Anaxine VSA, and we use the FACS. Here you can see the scatter plot showing that with increasing concentration, the apoptosis is increasing in the early and late stage, and in the graph bar also you can see that while increasing the concentration, of course the concentration was taken based on the IC50, but you can see that it's increasing in dose-dependent manner when we treat the MCF7 with compound 1 and 6. Of course, in breast cancer, you can, we know all that many pathways play a major role, like PI3K, MAP kinase, all these pathways play a really key role in breast cancer. We decided to initiate to study the MAP kinase because we found in recent studies or in the literature that it has an effect on the triple negative cells, which is the more aggressive cells. And we found that MAP kinase marker like phosphoric, ERK, and phosphomec and MEC also inhibited by the treatment of compound 1 and 6 on MCF7. The treatment was less than 24 hours, and we evaluate also the apoptotic marker to compare. And we use the classical apoptotic marker like BCL2, like MCL1 and BAX. We found that the treatment inhibit the anti-apoptotic proteins MCL1 and BCL, but it increased the bags which promote the release of bags which promote the apoptosis. So, based on our data till now, we believe that those compounds can disrupt the cell signaling in MAP kinase, and of course, the MAP kinase regulate the cell proliferation and cell growth. So therefore, it might induce the apoptosis in the cells. In conclusion, we have identified these 8 compounds or the series of compounds with potential anti-cancer activity, and the mechanism of action most probably work through the MAP kinase pathway, but we need to do more investigation and to confirm this. We would like actually to find or to confirm the target of these compounds, especially compound 1 and 6, and we will use actually the pull-down assay to identify. If we can identify the target, then we can take it to next level. We will get help from our collaborator, Dr. Mohammed Misgaldi from King's College in London. He will help us to establish this technique actually. It's very important to identify if it is a specific target or maybe it will be multiple targets. This research has been registered in the United States Patent and Trademark Office, and of course العمل لشخص واحد أو ريسيرشر واحد ما راح تجيب نتيجة لو ما كان في team work. So healthy science أساساً need team work. I would like to acknowledge كل التيم member اللي معايا في جدة University with this work أصلاً funded by TSI of جدة University. طبعاً شاكرين لهم. والتيم members اللي معانا من جامعة جدة، دكتورة رؤى الريني، دكتورة علاء الأحمدي، دكتورة ريما عتيبي، from King عبد العزيز University من Pharmacy College, Prof. عبد الستار, and from Egypt, Dr. مصطفى العربي, and from King's College, of course, Dr. محمد المزقلتي معانا, تحية له كمان, و طبعا Institute of Genomic Medicine for the technical support, Dr. Prof. Hadim Chaudhary, Dr. Hiba, Dr. Aisha, of course, Dr. Peter, and Sheikha, all the technician staff, and from King Fahad also Research Center, Prof. Anwar, Prof. Sam Azhar. نشكر أكيد الكل اللي تعاونوا في الريسيرش و thank you so much for your listening.
Thank you very much, Dr. Hind al-Khattabi. We would like to welcome our third speaker, Dr. Jihan Rahimi. Dr. Jihan will present next generation cancer diagnosis and treatment, innovation, nanotechnology, immunotherapy, biomarkers, and biotherapy. Dr. Jihan is an associate professor of microbiology and immunology at King Abdulaziz University. She's a member of Immunology Unit at King Fahad Medical Research Center. She graduated from the University of Sheffield School of Medicine with numerous publications and extensive supervision of master and PhD students. Please welcome Dr. Jihan.
Okay, Bismillahirrahmanirrahim. Thanks for the kind invitation and شكرا لحضوركم في هذا الوقت. I'm glad to present our lab work in cancer diagnosis and treatment, innovation in nanotechnology, immunotherapy, biomarker, and biotherapy. As a beginning, as a simple introduction about cancer. Cancer developed due to the accumulation of genetic and epigenetic alteration, resulting in uncontrolled cell growth and invasion of surrounding tissue. Cancer cells grow widely in bad healthy tissue and can spread throughout the body, leading to significant type of cancer. And as you heard from the previous presentation, cancer is a significant public health concern worldwide. Global demographic trends suggest an increase in cancer incidence over the next few decades, with 420 million new cases presented annually by 2025. In recent years, innovative technology like nanoparticle enhanced vaccination, immunological modulation, oncolytic biotherapy, and immunocheckpoint biomarker have transformed cancer detection and treatment. These novel strategies provide focused, personalized, and more effective options for cancer treatment while decreasing the side effects associated with traditional treatment. Traditional treatment, as they said before, like chemotherapy and surgery and radiation, which work directly removing or killing cancer cells, often have effective feedback, but also have a significant side effect. Moving to targeted therapy, which emerge as a more precise approach using drug to attack specific cancer cell protein. In contrast, chemotherapy immunotherapy. The immunotherapy is a game changer since 2013, stimulate the body immune system to recognize and destroy cancer cells. It's offered a long-lasting systemic effect and can be combined with other treatment and work across multiple cancer types, especially immunocheckpoints and CAR T cells. They have shown remarkable success in last-stage patients, even when their treatment have failed. So here I'm giving some examples of immunotherapy and the area that we published some work in it, and we work in this area. So I'm going to start with oncolytic virotherapy. Oncolytic virotherapy, like Newcastle disease virus, selectively kill cells through viral infection and replication, which promote an immune response against the tumor microenvironment. Immunotherapy, including the use of oncolytic virus, have gained traction in cancer treatment, aiming to enhance the body's anti-tumor immunity. So in this area, we published this work conducted by Prof. Dr. Zahir about Newcastle disease virus virotherapy, where we evaluate the dual effect of Newcastle disease as both an oncolytic agent and immunostimulator. We conducted a comprehensive in vitro and in vivo studies. So in lab, we used two kinds of colon cancer cell lines, were treated with Newcastle disease and virus and tumor cell lysate. MTT assay assessed cell viability, and also we used annexine to confirm apoptosis induction. In vivo, we used mice bulbs bearing colon tumor receiving treatment of Newcastle disease virus, T-cell lysate, or both, and post-treatment immune response were assessed using flow cytometry for specific markers such as CD4, CD8, CD83, and CD86, and ELISA to assess and measure cytokine level, such as interferon gamma, interleukin 4, interleukin 10, and interleukin 12 in blood and in spleen. So for apoptotic induction, flow cytometry results showed an increased apoptotic cell number post Newcastle disease virus infection, indicating that Newcastle disease virus effectively induced immunogenic cell death, as you see in these figures after 24 hours. And for phenotype profile for T cells, immune response was measured by flow cytometry analysis, and we revealed an elevated level of CD4 marker and CD8 for T cell, which indicate an enhanced immune response. And regarding dendritic cell, flow cytometric analysis revealed that the combination therapy induced by the highest expression of CD80 and CD86 on dendritic cell essential for T cell activation. By ELISA, cytokines level were measured, and we found an increased level of interferon gamma and interleukin 12 in the spleen of NDB plus TCL treated mice, signaling to robust immune response, while we find interleukin 10 were significantly lower in treated groups, suggesting a reduced immunosuppression. In conclusion, the research highlights the promising role of NDB as an oncolytic virotherapy for colon cancer, demonstrating significant anti-tumor effect and the potential for immunomodulation. Going to cancer vaccine, another type, and just a basic immunology. I say hi for my student there. I think they are aware about this basic thing in Bio 430. Well, we know about dendritic cell as a key immune cells in antigen presentation. So these cells act as a messenger, capturing antigen from cancer cell and presenting them to T cell. This presentation activates the T cells to recognize and destroy cancer cells. However, tumors are smart. They generate the environment that inhibit the activity of immune cells, including dendritic cell. This tumor microenvironment, or TME, may limit dendritic cell ability to activate T cell, limiting the efficacy of immunotherapy. So to overcome this challenge, researchers have been exploring various strategies to enhance dendritic cell function and delivery. One promising approach involved the use of nanoparticles. Nanoparticles can protect dendritic cells, improve their delivery to the tumor site, and enhance their ability to stimulate T cells. So in this area, we conducted this study by our master student, Najda Al-Atabi. In this study, we looked at the possibility of employing Shitosan nanoparticles to associate with dendritic cell and develop a more effective cancer vaccine. So Shitosan nanoparticle was studied in the lab by electrogenation with TPD and analyzed for size and zeta potential to confirm stability in immunodelivery. And bone marrow, as you can see, derived dendritic cell were differentiated using CSF and interleukin-4, then activated with LPS to promote maturation. Pulp mice were divided into groups receiving Shitosan nanoparticle alone, or dendritic cell alone, or dendritic cell associated with Shitosan nanoparticle to measure immune response by flow cytometry in blood and in spleen. And the result, as you can see, by flow cytometry in A and B, this is in blood. Dendritic cell associated with Shitosan nanoparticle group showed the highest expression of CD83, CD14, and CD11 marker for DC activation. MHC2 expression was significantly also elevated in dendritic cell and dendritic cell associated with Shitosan nanoparticle, indicating the strong T cell priming potential. And in the second row, C and D, you can see the in-spleen result. DC associated with Shitosan nanoparticle demonstrate the strong upregulation of CD83, CD11C, and MHC class 2, confirming the enhanced DC functionality. So in conclusion, the research provides compelling evidence that the dendritic cell associated with Shitosan nanoparticle vaccine can elect robust immune response in vivo model, offering a promising avenue for future cancer immunotherapy strategies. And this is under application, hopefully to be published soon. In another area, immunocheckpoints protein. Immunocheckpoints biomarker are proteins that regulate the immune response and can be inhibited or activated for cancer treatment. Important checkpoint protein include the BD1 and CTLA4, and they are well known. Immunocheckpoints inhibitors have transformed cancer treatment by enhancing the immune response against tumors such as anti-BD1 and anti-BDL1. So again, basic immunology about T cell activation. We know it's needs two signals. The first is antigen recognition, and this is by dendritic cell presented the antigen by MHC to T cell receptors, and the co-stimulation signal by the checkpoints protein. Tumors suppress this process through immune checkpoint, but checkpoint inhibitors can restore T cell function and enhance anti-tumor immunity. So in this published work, and it's conducted by our master student, Mahi Youssef, we investigate the changing level of immune checkpoint protein in the serum of breast cancer patients. So we collect blood from breast cancer patients and compare it with healthy control using multiplex bead array for certain biomarkers such as BTLA, M3, CD28, and others. And the results reveal some biomarkers such as BTLA, HBM, toll-like receptor 2, and BTL1 were found at a higher level in the blood of humans with breast cancer, while CD86 was lower. Here you can see the result for biomarker comparison between malignant and non-malignant serum sample from breast cancer patients. So in conclusion, according to this study, the biomarker BTLA, HBM, TLR2, CD86, and BDL1 might be used to monitor breast cancer and predict patient outcome. And more publication in this area. This publication conducted by Dr. Fatma Basingab and colleague exploring the HBM as immune checkpoint, and HBM as immune checkpoint that may suppress T cell activity in leukemia, making it a potential target for immunotherapy and useful biomarker for diagnosis and disease monitoring. And another publication also by Dr. Fatma about TIM3 as an immune checkpoint receptor found to be downregulation in BLL, especially in severe cases, suggests the potential role of this biomarker as a negative biomarker for diagnosis and diseases monitoring. And I can't end my speech without mentioning adaptive cell therapy. It didn't work in this area, but it's a promising area. It could be called also a cellular immunotherapy, where we use patient immune cells, like T cells, to fight cancer. The cells are collected, modified in the lab, and with special receptor to target cancer cell, and grown into billions. And then after chemotherapy, to prepare the body, the modified cells are infused back to the patient, where they attack cancer cell and multiply to help control cancer. And also CAR cells, now there are many trials to treat other diseases such as autoimmune disease. I would like to thank all members in our lab and everyone who contributed in this work. وأحب أختم بقوله تعالى بسم الله الرحمن الرحيم وما أوتيتم من العلم إلا قليلا. شكرا.
Thank you, Dr. Jahan, for the lovely talk. Our final speaker for today is Dr. Khalda Nasser, discussing AI-guided approach in cancer, a step forward in precision medicine. Dr. Khalda, an associate professor in molecular biology and medical laboratory science department. She's an admin and research member in the Institute of Genomic Medical Sciences. She collaborates in Prince Al Johara Center of Excellence in Research of Hereditary Disorder. Welcome to Tara.
Just wait.
السلام عليكم ورحمة الله وبركاته. اللهم صل على سيدنا محمد وسلم. Thank you, King Fahad Medical Research Center, for the opportunity to participate in such a bright, well-minded event with the group of scientists together. And I would like to thank all the successive partners with the King Fahad Medical Research Center to achieve the success of this conference. Today, I would like to talk about an overview of AI-guided approach that is transforming we all know the emergence of this technology is transforming the face of medicine. And I would like to focus on the application of AI in precision oncology, and I will highlight some of the unpublished work that we are currently working on in the ongoing project from King Abdulaziz University Hospital, recruiting patients of breast cancer. So this is the outline of my talk, an overview of AI application in oncology, and then the integration of machine learning in cancer target discovery, the model validation that we will be using for testing the performance of machine learning model. As some of you might know, the AI-driven cancer that when they are applied in radiological, and I'm sure it has been covered, I think, in day 2 session, when they are using for the mammogram, the CT scan, and ultrasound, there are some features or morphological characterization that they are looking at. It's called radiomics. These markers are used to to describe or differentiate the malignant cells from the wild type or normal cells. One of the popular techniques is Google DeepMind, have demonstrated and outperform other tools for increasing the accuracy of detecting malignancy from radiological images. Also, AI-driven pathology, the histopathologists, when they make grading for the tumor or specify the stages of cancer, depending on the morphological features and some biomolecular markers that are expressed at the surface of the tissue. There is also a lovely tool that has been used recently by the scientists. It's called Path AI. It's accelerating the machine learning model to look at all these slides, nice fancy stain slides from the immunohistochemistry in order to differentiate again the prognostic, the progress of the cancer and the prognostic information for the patient, whether they are responding well to the treatment regime applied by the clinician. This is the slide that shows AI-driven. It's again an overview how the AI-driven treatment plan of precision medicine. When you hear the word or the term precision medicine, comes to your mind. It's an individualized treatment depending on each individual characters. So, what is the main driver or the main engine of our differences in terms of treatment response, in terms of diagnostics? It's the genetic profiling. And there is another term, fancy term, that are recently used by the scientists in precision medicine or personalized medicine, is the multi-omics integration, where we rely on genomic profiling and the expression of these genes that are different between cancer patients, and will highlight or key specify the treatment regime plan for each of these cohorts. Radiotherapy optimization, one of the ideas is that the patient might wear a wearable device in order to see how much of radiation therapy is being absorbed by the patient while they are going under radiation therapy. And there will be images that are provided pre and post treatment. And each individual can be categorized and offered specific treatment regime depending on all these integrated data. The AI-driven target discovery, we have again drug repurposing. AI now has shown the acceleration of the target marker that is already been published and are already used in cancer therapy. Some patients might be responding very well to the treatment, and some patients might demonstrate side effect. Again, the genomic profile background. This work has been already proposed and applied through RDIA, hoping to get funded soon, to integrate all these multi-omics with radiological and histopathological images for treatment planning. And the target identification as well, because when we do genome sequencing and transcriptome analysis, proteome and metabolome, we can identify first with the genome the underlying molecular or genetic novel marker that differentiate the patient responses from each other. At the proteomic and the metabolomic level, that shows quite evidence for precision medicine, the functional of these targets and molecular biomarkers integrating together and the interaction for that one. This shows the predictive oncology modeling. Actually, this image I adapted from a research paper. As you can see the link here, the reference. It's very nice holistic approach work that has been done by a group in US for the predictive modeling, where you can see when you say prediction for oncology, you're talking about patient survival, you're talking about the diagnosis and the prognosis of the patient and the stage of the tumor. And when you build the model, integrate data, genomic and transcriptomic data along with the images, you can increase the accuracy of the sensitivity of your prediction for the cancer model. In vitro lab experiment, this is again the work that will be done once we get funded, hopefully, with the collaborating parties in this research, King Fahd Medical Research Center, And we are aiming to, once we successfully apply the model and we validate its performance at the mathematical and technical level, then we will translate it into in vitro analysis using human organoid stem cell, hopefully. And this is the platform for the AI-driven approach, where you can integrate the omics platform, as I mentioned, the proteomic, the metabolomic, and the genomic, integrated with the targeted drug delivery strategy that is currently proposed. And one of the things I couldn't share in this slide because it's unpublished yet, and for the sake of time, we have managed to download a huge public data set from bladder cancer that is available online from the gene bank, and we apply the machine learning model to try to identify the significant associated genes from the RNA sequencing analysis. And we found like 5,000 genes out of 500 top genes out of 10,000 that are mostly involved in the pathway of the progression of bladder cancer. However, this work is limited by the data set structure. It was only, I think, 500 cohorts. The more data you feed the model, and the more training the model, the more accurate the testing data set will come and provide you more accurate data. So this is one of the proof of concept we are aiming to publish soon, hopefully. And this is just challenges and limitations of generally the AI applications now with the ethical use, the data privacy, the data governance, multiple stakeholder collaboration at the national and international level must be addressed in order to facilitate accurate data handling and not to be released without proper security measures that be implemented. And future development for for example, the radiation oncologists in the hospital, they're using, the doctors and the chef that reading the mammogram, reading the images. There is an imposing bias when it comes to the eye of each individual depending on their skill background. Whereas when you apply machine learning and advanced image analysis, you could overcome that human type of bias. It will help us also to improve the prediction and enhance the decision clinical support system for the patient. The take-home message, and this is the final slide, we are hopefully at the era of that AI is accelerating the research and innovation in biotech and cancer therapy and diagnostic. And hopefully our project that we talked about looking at breast cancer, once it gets published, I will present the model development outcomes next coming meeting. Ethical and data privacy concern must be addressed carefully. And one more key thing when we talk about precision medicine with cancer or any chronic disease or any rare disorder, it's worthwhile noting it's population specific. The molecular epidemiology is different from Arabs and compared to the West, so we need to be careful with trying to sequence the whole genome from Saudi Genome Project, for example, for instance, and establish centralized database. Hopefully, I know it's challenging at the moment, but hopefully we are heading toward that path. This is the research team that we are working in the breast cancer project using AI. And thank you so much for your attention.
Thank you.
Thank you, Khalida. So I would like to call on the speakers on the stage for a small discussion. Now, the floor is open for questions. We'll be happy to take questions, please. If you have questions, go ahead and ask your questions.
Any questions?
Assalamu alaikum. My question is for the first speaker. She mentioned that they tested their compound against MB signaling bus. How do they get this idea that the compound can have effect on this target? So,
Sorry, I think we missed the first speaker. I'd like to respond on behalf of her. So usually what happened when you see the slides, she was trying to convince the audience that breast cancer come up with some certain going up. You know, one of them is PAC kinase, which is playing a big role in the cancer progression. And apart from PI3K, you know, there is a mutation in PI3K pathway. So that was the reason for her to choose PAC kinase. That I believe that was the reason. I believe that what she said in the presentation. So I don't know. I hopefully answered your question or not.
lots of pathways.
Yeah, I know lots of pathways, but you cannot target all the pathways. You have to see the most significant pathways which has been noted in that particular case. Okay. Any other questions?
Thank you all for the great presentation. I have a question to Professor Daham. So you mentioned that you see EV elevated in COVID-19, right? Yeah, I wonder if you looked at thrombosis in these patients, are there any association between these microvesicles and thrombosis? Can they trigger the clot formation?
The work that we have published just fully focused on the untargeted metabolite, on the exosomes. Actually, we have not looked at you could remember we only worked on the metabolite. We only did full metabolite analysis of the exosome from the COVID-19 patient from the mild and severe and ICU. We have not looked at this.
I have a question for Professor Pilzer. I would like to know more about the extracellular vesicles in myocardial infarction. Do you know what the contents are and what they are targeting?
Actually, this is not the area we are focused in our research, but the work that I have highlighted in my last slide was really interesting. It showed that the myocardial fractions patients, exosomes carrying some important molecules such as proteins, lipids, microRNA, and also metabolite. But I am not interested about this area. But what they have shown that they can increase the productions of EVs, which can improve the health of the patient.
Since we are talking about exosome, can I ask you something, Dr. Clever? Now, with the current advancement in commercial product and anti-aging product, is there any scientific evidence supporting that exosome have any anti-aging effect? And is it safe to use them?
Actually, currently, there is no FDA-approved product in the market. However, there is a lot of evidence supporting that exosomes can play an important role as an anti-aging. But unfortunately, there is a big numbers of beauty clinics that are selling exosome products, including in Saudi Arabia. And even in the United States, there are more than 500 clinics that are selling the exosome products and stem cells products. And when you ask those doctors or those surgeons, they claim that it is surgical procedures. But currently, as I said this morning, there is no FDA product approved from exosome.
Any more questions? So if not, then I have questions for all three of you, one by one. I'll start from the left. So, John, I would like to know a little bit more about these oncolytic viral therapies. So, did you test it, and did you see other virus-based therapies? Apart from Newcastle virus, there are other potential viruses that can be used for this purpose.
Yes, this oncolytic virus was currently provided by Dr. Zahar. He's conducted this study. Maybe in future we want to look to another oncolytic viruses and another type of tumor, inshallah.
For Dr. Khalid, so I would like to take this opportunity because we are talking about precision medicine and we're talking about a lot about AI. And being you affiliated with the Institute of Genomics here, and it is like a recommendation I would like to give from my side that we have a lot of students sitting here. So I prefer that all of you will think about learning basic R and Python. It's very important that in these days, if you are a clinician, non-clinician, if you are a grad student in basic science or maybe applied science, you must learn basic R and Python. It's very important. It is not going to, it's not very deeply convicted. There's a conviction about it's a very hard thing, but it's very, very fairly easy. And it is going to give you a lot of power in terms of analysis. And, you will see a lot of scope of learning these things. And what do you think in your institute? Do you plan that with the graduate students coming up, you have a course designed for it?
Thank you for this question. It's actually one of the challenges now that students they're going deep into data analysis and mining the literature and all of that. That takes quite a lot of time. If it's not handy for them how to learn the basics of how to use Python or like the basic data science, the algorithms and all of that, that would be a bit of challenge in the near future, I would say. In our institute, we have a department called Bioinformatics and Artificial Intelligence. Me and Dr. Reem, Dr. Silami, Dr. Noha Reyes, Professor Jad, Dr. Peter, we are working on course specification for a master program involving basic AI, advanced AI, and how to incorporate this. If you say AI, eventually a tool, people have it. It's a big theme where it can be incorporated in any of the medical applied sciences nowadays. So hopefully, once this program, gets approved by the Deanship of Graduate Studies, we will be ready to have postgraduate students and teach them the necessary learning approach. Hopefully.
Thank you. It's a great initiative. Yeah, please.
I think code writing, programming, it is one of the key skills that the future biologists, future biochemists need to learn.
If you allow me, we are biologists and scientists, medically purely, so I have no clue about computer, but there are amazing online virtual courses that are done by MIT, Harvard, and it's paid courses, of course. And King Faisal Specialist Hospital did a course called AI Developer Program, which teaches us from scratch how to use Python and coding. And I took that course for 5 weeks with a certificate. After that, I learned a lot about AI and machine learning. So maybe at the beginning, the student will feel like this is not my skills, this is not my background, but I would really recommend, attending such courses to improve the skills.
That's it. That's what I believe. One thing has to be done is to do a workshop, from your side, to inform the students about it, that these courses are freely available and can be accessed very easily around. Thank you. And Dr. Professor, I have a question for you. And I would love to know more about plant-derived EVs. What are their future in your eyes?
As I mentioned in my presentations, the mass productions of stem cells exosome is a big challenge in the field. So now, since we find the same component or the active component in the exosome, that we can find it in the alternative resources such as the plant, it will be a big future in the field. And I witnessed that the last, it is the last, and first and last time I attended
the conference there in Dubai, which is focused on the beauty and the cosmetics products. Plant exosome is in every corner in the conference. All people is talking about plant exosome as a future product. So I have total belief in the plant exosome as a future.
Thank you. No more questions? Please.
Actually, we got this question from the online section, from the YouTube. One of the viewers asked Dr. Faisal, and the question is, from where did you isolate the mesenchymal stem cell, and why did you choose this source?
It's from the bone marrow of the mouse. Yeah, because it is the most rich source of stem cells and much easier way of isolating the stem cells.
So, any more questions? If not, we would like to conclude the session, and I would like to thank all the presenters for their kind efforts. Special thanks to you in the whole session for me. And I would like to thank you to all of you who've been here, showing your interest in our talk. And I would love to see next time when this happens, one of you come and start presenting your work here. So looking forward. Have a nice day. Thank you for coming. Wishing you all the best. Thank you.
Lots going on here. Mediators.
شكرا جزيلا.
يعطيكم العافية.
We will be moving to the poster awards دحين إن شاء الله. دكتور تركي يكون معنا. From the third position فازت فيها الطالبة أفنان عابد الفضلي. عندنا أفنان في المسرح. موجود أفنان.
Second place.
Second place للطالب عبد الرحمن الأحمري. Prime Minister of Islamic Sustainability for the Agricultural Factories. عبد الرحمن موجود؟ موجود عبد الرحمن. First place راح لندى الشلالي detection of pathogenic mutations in genes inside breast cancer patient using whole exome sequence. ندى مولودة ولا مين موجودة؟ مولودة. يلا يا ندى. خلاص لقينا ندى عشان كذا كنا ندور فيه. والله هيكون راح.
Thank you all for coming.
We conclude our session today. الله يعطيكم العافية أجمعين. شكرا لكم ونشوفكم بإذن الله غدا.